sir, deans, heads of the various departments, directors, and my dear friends. I cordially welcome the participants, those who are making this event to be a successful one, stars of this conference. About the conference is about our country. It is blessed with a vast and varied marine resources. The mariculture is the rearing of aquatic organisms under controlled or semi-controlled conditions in coastal and offshore waters where the salinity is maximal and not subject to significant daily or seasonal variations. Mariculture contributes to the production of protein-rich food and has been source of livelihood of millions of coastal villages. The global annual production from the marine environment accounts 21.6 million tons. In 2020, mariculture contributed to 91% of the global total production, both marine and brackish environment. India's mariculture industry produces more than 5 million metric tons of product annually, valued at 350 billion rupees. Aquaculture must bridge the gap between sustainable fisheries and demand, and it must do so in a sustainable way, with sustainable feed raw materials and minimal environmental impact, and it must be recognized and accepted as a valued provider of excellent food. In this regard, the Department of Marine Biotechnology, AMET, organizes the National Conference on Mariculture in India, recent advancements. Well, I don't want to take much of your time. Our eminent professor, President, Indian Association of Aquatic Biologists, and convener of this conference, Dr. K. Altaf sir, will brief out the theme matters. Once again, a warm welcome to all the dignitaries on the screen and off the screen. Thank you all for giving me this opportunity. Let's learn together. Thank you.
Thank you, sir. May I now invite our eminent and beloved Professor Dr. K. Altaf sir from Department of Marine Biotechnology to deliver the theme address. Over to you, sir.
Thank you, Sarna. Good morning to all of you. Most respected Vice Chancellor, Colonel Dr. G. Tiruvasagam, respected Registrar, Dr. Jayaprakash Vail, respected Controller of Examination, Dr. Muthirilan, Dr. Shaul Ahmed, Dr. Kailasam, Dr. Santanam, Dr. Edward, Dr. Mayakandan, resource persons of this conference, my esteemed deans, directors, HODs, and members of staff of biotechnology department and other departments, respected Shama Madam, Secretary of Indian Association of Aquatic Biology, and my dear friend and HOD of Zoology in New College, Dr. Asrar Sharif, esteemed colleagues, it is my privilege to just say a few words about the theme of this conference. When we talk about humankind or mankind, we always see there is quite a lot of variation in different countries pertaining to the availability of food. And this, in some countries, underdeveloped countries, it's a big issue. And about this, there is increasing consciousness, even in the international bodies like WHO, Food and Agriculture Organization, UN assemblies. And if you just try to look at this problem, how to provide quality protein, protein deficiency is the important aspect which cripples many lives. Aquaculture sector ensures future good health and food security to the entire population. India is blessed with, as Dr. Silvinathan has been pointing out, a vast coastline of 8,000 kilometers and also exclusive economic zone of about 2.3 lakhs square kilometers. And so we have a very ample and vast area for aquaculture process. The potential is very high. And this exclusive economic zone, EEZ, provides real a kind of resource that is through capture fishery or aquaculture, we can have a kind of economic growth which can be on par with any other developed country. But unfortunately, now, even with regard to the mariculture, it is only 15% of the coastal area alone is under practice. There is no thought about venturing into the offshore fisheries. It is all only the capture fisheries existing so far. But this offshore culture or deep sea culture is something which is much better than all other system of aquaculture. If you just try to look at the aquaculture itself, it is started centuries back by Chinese, by say it is something like where it is a land culture. What I mean by the land culture is the rice fields, when they get inundated, the Chinese introduced the crops in their rice field and cultivated for some time. And that is the origin of the aquaculture. So from the land, that is the rice field, we have gone to the ponds, lakes, rivers, which are happen to be the a separate assembly of organisms, and with the natural food, they grow them and then utilize them. So from land to freshwater, then freshwater to the coastal water, and we are in the coastal water. What exactly is the present status of the coastal water aquaculture already we have seen. But we did not venture into the real mariculture. That real mariculture is there in the open sea, in the deep sea. And what are the challenges with regard to this? Again, even if you just take this freshwater aquaculture, it started with the carp, and thereafter the catfish has also been introduced, and the hypophysation technique with which we can artificially produce the seeds revolutionized the thing. And this is the one important reason why even today we can get in the market a fish for about 50 to 60 rupees a kg. You could see many of the vegetables are much higher priced than this. But the kind of protein which the fish provide is much superior to even the vegetables. So this is how we could see that there is a good revolution in the freshwater culture, and this has led to a blooming of carp culture. But why don't we do that with regard to the marine culture? This is the vital aspect. When we think about this aspect, we can, with guarantee, we can say marine fishes are much better than freshwater fishes. Marine fishes are much better than even the organisms or animals formed on the land. You take poultry, or you take the pigs or piggery, or if you take the cattle farming or anything, all these farming converts the feed which it takes into a very less quantity of their flesh or meat. For example, in the case of the pigs, it is 4 kg. In the case of the cattle, it is around 10 kg. 10 kg of feed can be converted to only 1 kg of meat. While in the case of the fishes, it is 1.2 kgs of the feed which can be converted to 1 kg of fish flesh or meat. Look at the difference. This is the very vast, important difference. And so more than the land cultivated organism, fish is better. Even with regard to the freshwater fish, it is much more important to culture marine fish. And what is the reason? Say, marine fish, in the case of the freshwater fish, some adaptations are there which require more metabolic processes. But in marine fishes, it is much lower. Even with regard to the land fishes, in the land organisms like sheep or goat, most of their energy goes into the production of the hardy skeletal material like bones. That is why there we have less meat production. Fish has got the minimum bare required of requirement of the bones. That is why more flesh is there. Further, there is no gravitational pull for which quite a lot of energy has to be spent with regard to the marine fishes. It is a very ideal condition, no climatic drastic changes. So everything is meant for it. Probably this medium, if you just try to look at the saltwater medium, where the life is originated, it is something very, very, what do you call, ideal medium for the life to sustain. So from that point of view, we have uh, started culturing the organisms, and we could find some solutions. So once when we have ventured into the land to freshwater, freshwater to the coastal, and this entering into the marine fish is not ventured so far. What could be the reasons? There are quite a lot of things. For example, if we take the infrastructure, the regulatory acts, the kind of seeds available, all with regard to all these things, there is immense problem in mariculture. In the case of the freshwater fishes, the size of the egg, fish egg, is much bigger compared to that of the marine fishes. As a result, we cannot culture them easily, or we cannot produce their seeds in the hatchery the way in which we produce the seeds of either catfish or the carp in the case of freshwater fish. Nevertheless, if we consider the economic point of view, a catfish or the carp can give you only 50 to 70 rupees per kg, while marine fish will give 700 to 1000 rupees per kg. There is a great demand for tropical fishes, tropical shrimps all over the world. Even in the 80s and 90s, per kilogram of shrimp, more particularly the Penaeus monodon, has been sold for 700 to 800 rupees on the banks of the farm. So that is the advantage of the marine culture or mariculture. And so this culture, mariculture, has got some very important challenges. And first challenge is availability of the seeds, as I said. And this availability of seeds has been from the Indian context, there has been concerted effort by the CMFRA and SEBA. And we have in our program today, Dr. Kailasam, who is an expert in marine fish culture. And they have solved the problem of breeding or raising the brooder stock, then making them to spawn and producing the eggs. And there is quite a lot of bottleneck as far as the rearing of the larvae is concerned. And this is something, again, very important aspect. Live feed has to be there. And this live feed, that too indigenous live feed, if we have that indigenous live feed, then it is a natural feed so that we can grow them better. The seeds will be of quality nature. There won't be any kind of susceptibility to diseases and other things. So that way, feed is very important. In our program today, we have Dr. Santanam, who is making excellent work on the live feed culture, more particularly the coffee pot culture, and also Dr. Mayakandan from our own institution, who is an expert on the microalgal or algal culture. So this is the second aspect of it. And thirdly, with all these things, if the environment is not protected, always either shrimp or fish is highly susceptible to pathogenicity and diseases. And we have in our in the midst of us Dr. Shaul Amit. And Dr. Shaul Amit is one of the world reputed researcher on the prawn and fish diseases. He started his career as, or even as a scholar. He did in 1980s or late 70s research on the larvae of the monodon prawn or shrimp. And then as a scholar itself, he had several papers in the Aquaculture Journal where it is very difficult to publish. He is an expert. We have the fortune of having here. And he has brought about some many products with which we could now cultivate the Benami without any disease problem. So these are the resource persons, and when all these things is there, we have to have a person who has to tell how a hatchery has to be constructed, what kind of biosecurity has to be taken. And to attend this, we have Dr. Edward Danish. He is the one who has designed many hatcheries. He himself was the owner of a hatchery, and he having a very wide knowledge in the hatchery development, and also he's a consultant in many organizations. So the conference is packed up with this team. So the technology, I said, the technology regarding the breeding and also the larval rearing is over, but still we need to go for selective breeding, enumerating more number of fishes. And also we should have to have genetic improvisation with which there can be better aquaculture. Then the second is mass production of indigenous feed, and we have not made any effort in this. There is a big lacuna in this. Thanks to Dr. Santanam, he is taking quite a lot of efforts in bringing about the density of the culture which can suit the hatchery requirement and commercial demanding. So that is also need to be attended, and we have to have the biosecurity aspects. That is again is a challenge. And finally, we should start offshore culture, deep sea culture. It requires quite a lot of infrastructure. It requires quite a lot of thinking about engineering and putting up a farm in the mid of the sea, under the water. And it is not that somebody has not done it. US has done it. The Salma organization, a company, it has put up such a structure, and in a matter of 4, 5 months, 6 months, they have produced 1.5 million eggs in one particular farm without any problem. So that is the scenario. They have done it in 2017. China has now established so many deep sea farms producing huge amount of fishes. Norway also ventured into it, and it is a high time for India also to venture onto it. And because it gives lot of money, I think no agriculture or no, even in the producing a movie may not give so much of money, as much of money if we invest on this deep sea culture or mariculture. And this exactly is the purpose for which this organization is, our organization has organized this program in collaboration with the Indian Association of Aquatic Biologists. And I hope we have the right people to discuss about these aspects. And also there are many participants to present their papers. So with this, let me conclude, and thank you very much.
Thank you, sir. May I now invite our beloved registrar, Dr. M. Jayaprakash Will, sir, to deliver the inaugural address. I welcome you, sir.
Thank you, Sona. Most respected Professor Altaf, actually the driving force of our department and the intellectual front. Very good morning, sir. And most respected guest speakers, Dr. Shahul Hamid, Dr. M. Kailasam, Dr. Santanam, Dr. Danish, and our own colleague, Dr. Mayakannan, and respected HOD of the department, Dr. Sendil Nathan, faculty members, research scholars, and more importantly, all the participants. A very warm and good morning to all of you. It is really indeed a happy and pleasure for me to inaugurate this national level virtual conference on mariculture in India, recent advancements, in association with the Indian Association of Aquatic Biologists. I profusely thank the office bearers of Indian Association of Aquatic Biologists for having come forward to join their hands with our university to conduct this a wonderful conference. After having listened the elaborate theme address of Professor K. Altaf, everyone must have understood what is the immense importance of this conference, because we are in bare need of alternative protein sources. When I look at the speakers who are all actually working on the exact field, the department as well as the organizing committee has chosen nice speakers who are having practical and year-long experience in the field. To be very much felt in this conference. And lastly, our own colleague, Dr. Mayakannan, my long-time friend, and colleague Dr. Mayakannan. He is expert in algology. He is actually a botanist by education and specialized himself into a algologist. He was trained by one of the world-renowned algologists, Dr. Anand. He is having the legacy of he is taking forward the legacy of Professor Anand. When I was a scholar at Madras University, I used to visit his laboratory very often, and I used to see the enthusiasm how Mayakannan was working on the taxonomy of algae. He is excellent taxonomist, both in terms of algology as well as plants. Excellent taxonomist. And after the completion of PhD, he has taken a different route. He went to work in the industry, and he is a consultant of so many industries, developing many practical projects and working with many industries. That is really excellent. Industry exposure is always very good. We are fortunate to have him in the department, and his presence will be very much useful for this conference as well. So when Professor Altaf was telling about the protein requirement, I would like to mention it as protein demand. Actually, we are in protein demand. Protein is very much required for the day-to-day diet of any human being. The people says that almost 0.83 gram of protein is required per kg of body weight per person per day. So a person, if it is 60 kgs weight person, he needs 60 into 8.8 grams. That much amount of protein is the daily requirement. So the protein also should be a quality protein, and there comes the protein demand. Our population is ever increasing. We could not control the population explosion. If the population is going on higher and higher, and the agricultural lands are getting down and down every year, and still we have large scope for marine environment only for meeting our protein requirement. If you look at the protein sources, mostly they are coming from cereals, meats, dairy, and of course from some insects also, and algae and fishes are the major protein sources. But if you look at the quality of the protein, as Professor Altaf sir said, quality of the protein, fish protein as well as the seaborne animal protein are having higher value than any other protein. The poultry is now occupying a major proportion of meeting our protein demand, but still it has many points to be considered. Excessive use of banned chemicals like steroids and antibiotics is causing a great concern for the usage of poultry. So in that case, this aquaculture or mariculture at a large scale. Mariculture is having a great scope, and it is our most expected technology to meet the protein demand. So in this context, the department has rightly identified the exact requirement of the society now, and the exact advancements from the actual field of mariculture has to be discussed in detail. So this department has organized a one-day event on mariculture in India, recent advancements. I congratulate the department head as well as the organizing committee for having chosen this very important topic. And also more importantly, the speakers, really they are excellent speakers. I have witnessed them in many occasions, and really the participants will be having a wonderful and very productive day by attending this conference. I wish the conference a great success, and I also wish all the audience as well as the participants to have a more productive day. Please stay the entire day. You will get more and more than what you expected. Thank you.
Thank you, sir. Now we'll start the first session, and I feel privileged to introduce our first speaker, Dr. M. Kailasham, Principal Scientist, Finfish Culture Division from Central Institute of Brackish Water Aquaculture, Chennai. He has done his PhD from CMFRI, Kochi in Marine Science. He has 27 years of experience in fish breeding farming and live feed culture. He has completed 7 major research projects and 4 consultancy projects from major funding agencies. And also, he has received 12 awards from different government recognized bodies. He has published 160 research articles in reputed journals with 852 citations. During his research period, he has developed standardization of breeding protocols and seed production technologies for brackish water finfish. With this short introduction, now I would like to invite Dr. Kailasham sir to deliver the lecture. I welcome you, sir. Over to you.
Thank you, madam. Good morning, one and all. First of all, let me congratulate the Department of Marine Biotechnology, AMET, for conducting such an important and very useful topic. It's very much required as on today. This particular virtual conference is also conducted along with the Indian Association of Aquatic Biology. And the topic Mariculture in India recent advancement is a need of hour because now there are a lot of concern about the food, about the healthy food and the food requirements for the growing population. And these are some of the aspects because in agriculture there are some issues are there, especially because of it all depends the monsoon, climate change, and all those things. But in fact, the source, the marine sources, especially the aquaculture resources, we are yet to establish. We have to establish in a bigger way so that we can get a better revenues. So in this regard, my topic today, I am going to present on brackish water finfish farming in India, challenges, opportunities, and way forward. Can you put a full screen? My PowerPoint. Okay. Yeah. Okay. Then you can just slide over. Next slide. Yeah. So if you see, we go to look into the global population. When we propose a project, when we take up any research problems, we go to look into broader perspective. Who are our target groups and what purpose we are taking the research projects? So in this regard, if you take in 2000 by 2050, global population going to be increased about 9.7 billion from the present population of 7.7 billion. So in this, there are about 795 million people are still suffering from hunger, and more than 2 billion people are malnutrition deficiency as per the FAO 2017. So food demand definitely is going to be increased anywhere within 9 to 58% by 2050. So FAO already estimated that the 22% of the Indian population is undernourished. That means that they are not getting a proper food. That is the FAO estimation. And if you look at all the food producing sector, agriculture or livestock or even poultry or even milk production or any sector, food producing sector, aquaculture is the annual growth of about 8%, which is the highest when you compare to all these sectors. Next slide. So in this regard, aquaculture has a very good potential that we can contribute a lot because India has such a rich resources. Next slide. So the rich resources in terms of freshwater, marine, and brackish water resources, and also we have a lot of species, candidate species in terms of finfishes and shellfishes. We have a lot of candidate species, cultivable species, economically important species, and these species are shellfishes or finfishes can be utilized if we have a proper seed production technology as well as the farming technologies. Next slide. Yeah, if you see the per capita fish consumption, Fish consumption, if you see 1961, it was about 9 kilo per head. But by 2015, it is at 20.2 kilo. So that means that about 1.5% is increase every year. The preliminary estimates in 2016 and 17 pointed out that it is going to be increased further around 20.5 kilo per respectively. And fish is considered as a healthy food, cheap source of protein, because if you go to any doctor, they say that you can have a fish from children to adult, because fish is having a healthy fatty acid that is really good for heart, and they propagate that fish is a healthy fish and healthy food and one can have it. Next slide. So in this point of view, the food demand, the fish consumption demand for fish is going to increase in the coming years, and definitely because we compared the few years back, the purchase power from the people, from the consumer is not that good. But in the recent years, because of the income is good, then the people are able to buy the fish, and there's a demand. There's a good demand for the fish. So definitely the demand for fish consumption is going to increase in the future because of its nutritive value, and aquaculture, definitely the growth rate is going to increase, not 8%, more than that in the coming years. Next slide. So this is if you see the world fish production, and this is the graph, and this is a trend you can see. So there is below, there's a pink color, you can see almost, it is the capture fisheries. The capture fish almost, it is not growing much, because it's almost, it reached to the, that saturated position. But if you see the blue color, that is aquaculture production increasing. Especially from the 90s, the aquaculture has surpassed, aquaculture production has surpassed the capture fisheries production, which clearly indicate that the fish production through capture fisheries is not going to contribute for the maybe just in static condition, but aquaculture is a scope. So aquaculture is the only scope where it can contribute the growing demand for the fish consumption for the global population. Next slide. Next slide. So what is the fish production in India, in the Indian context? If we really say Indian context, 2017-18, it is estimated that about 12.6 million metric tons the fish production, out of which 65% is from inland. This is mainly from the aquaculture production. Marine sector contribute only 33%. And we are able to export about 75 countries and about 13.77 lakh tons in terms of quantity, and value is about 43,000 crores. And it could earn a good amount of foreign exchange as a revenue. And if we see the overall exports, aquaculture fish products itself contribute about 10% from the overall exports, whereas agriculture exports around 20%. And overall to GDP, nearly 1% contribution from the fishery products. And if you see what are the states, the top 5 states fisheries, and if you see Andhra Pradesh is the maximum production coming from both marine as well as inland, contributes about 34.5%, followed by West Bengal 70%, Gujarat 8%, Kerala and Tamil Nadu. So these are some of the top 5 states which contribute the fisheries production in India. Next slide. Coming to the shrimp production in India, as far as the brackish water aquaculture is concerned, so the brackish water aquaculture is mainly with the shrimp species. And previously we were doing it monodon, but in the recent, for the past 10 years, we have been doing vanami, that is a species majorly cultured, farmed in the coastal states. So Andhra Pradesh is the maximum production. This is 2019 production data. Andhra Pradesh is about 71% production is there, followed by West Bengal, then Odisha, Gujarat, Tamil Nadu, and almost 8 lakh tons. 8 lakh tons as per 2019. The shrimp production is about 8 lakh tons. And of course, there are several issues. There are some disease issues, then FCR issues, then price fluctuation, feed cost, input cost, all these issues are there. But various organization are working how to come across these issues so that the production can be achieved in a cost-effective basis. Different institutions are working on this. Next slide, please. Okay, coming to the present, what are the infrastructure facilities available in the brackish aquaculture sector right now? If you see, there are about more than 600 hatcheries. Shrimp hatcheries are available in India, so they are able to produce about 125 billion seeds. And feed mills, there are a number of feed mills are available with the production capacity as on today 2.5 million tons. And processing plants can handle about 1 million tons of fishery products. And right now the farming area is around 1.76 lakhs. That's 1,76,000 hectares. Right now the farming sectors, and traditional, apart from that, traditional is around 40,000 hectares are already in the traditional this thing. And total potential area is around 1.2 million, 11.5 lakhs. Next slide, please. Next slide. Okay, what are the real issues? Previous slide, please. Previous slide. Previous slide, yeah. So what are the real issues? If you see, we are depending on a single species. So single species depending any farming activity is really risky, because if any problem comes, like in terms of diseases or anything, price fluctuation comes, then we may not have alternate species for farming. And already there are some issues I already mentioned, because there are environmental issues are there. Then keeping the quality healthy seed as an issue. Then feed alone contributes about 60% of the production cost. So cost effective feed is another issue. Then culture management systems. Then market fluctuation, international market fluctuation always, is not steady. A lot of other issues are there, marketing channel, everything is an issue. So in this context, we go for species diversification. Let us try out other species, other cultivar species, so that the farmer not only depending with the shrimp, but they can also take up finfish species as on the basis of crop rotation. One crop they can do shrimp, another crop they can do fish, and so that they can get a better avenue, better income they can get. And this will definitely help to achieve the sustainability. We have been discussing a lot about the sustainability, and this will help. In this context only, our institute SIBA has taken up candidate species, some important candidate species, brackish water finfish species, to develop a hatchery technology as well as a farming technology, so that the farmers can make use of these species also in the culture side. Next slide, please. So these are some of the species we have taken up in SIBA. So Asian SIBA, Lattice calcarifer, I just read out some of the important species we have been working. Food fishes, these finfishes can be categorized into two types. That one is the food fishes, and another one is the ornamental fishes. So food fishes are the seabass, milkfish chanos, Feldspar Atrophis rotensis, then catfish Mistus bulio, grey mullet Mungil cephalus, manger red snapper Budana subjectimaculatus, rabbit fish Siganda javus, then hilsa, ornamental species spotted scat, silver mooney, ornithochromite, and Canarin feldspar. So these are some about around 10 species we have been working to develop hatchery technology as well as the farming technology. Then we are also transferring the technology to the farmers in terms of demonstrations, in terms of seed supply, in terms of training, as well as the consultancy. Next slide. Okay, let us see one by one the Asian sea bass Lattice calcarifer. Lattice calcarifer is a popular fish in India. If you go to any state, this fish is very popular, and everyone knows, consumer side they know very well. If you go to even Calcutta, it's called it Bedki. And if you come to other states also, there is a local name is there. So this fish doesn't have no need any popularity among the farmers, because already popular fish. And this particular fish is having a very good growth rate. If you stock around 100 grams in the growth pond, within 6 to 8 months, you go to 1 kilo. Rapid growth rate, you can accept the pellet feed. And more than that, complete hatchery technology is available. A technology packages for seed production as well as feed production also is available. Then health, we can also stock a healthy seed. In terms of we can screen out any VNN, any viral or anything that we can screen out, and we can stock a healthy seed so that we can get a assured crop. So complete technology package is available for this fish, for this particular fish, in terms of seed, feed, as well as the health management. Next slide. So our main intention is that a land-based restaurant model. So any farmer or any entrepreneur, if they come, we propose this model. A farmer can take up hatchery or nursery for fingerling production, or even grow culture, and it can be also marketed. So the land-based hatchery to restaurant model we are propagating, and in a hatchery we used to supply about some 25 days old fry, about 1 to 2 centimeter size seed we are able to supply. From this seed, the farmer can rear to fingerlings in a small way, in a small hoppers they have according to their capacity. They can produce a monthly about even 10,000 fingerlings or 5,000 fingerlings, and each fingerling they can sell about 25 to 30 rupees, and even 10 to 15 rupees there is a profit margin for each piece. So that is a model we have been working for even for a small scale farmers or for entrepreneur model also. We have the customized technologies for seed production as well as fingerling production. Next slide, please. So these are some of the timeline where SIBA has done various activities over the years. If you see the first breakthrough, we are able to achieve in 1997. That was the first marine fish hatchery bred in India. My senior Dr. A.R. Tirunavasu sir, under his team, we have made a first breakthrough. We have started the technology. Then 2002, 2003, we have given the consultancy to RGC, Rajiv Gandhi Center for Agriculture, and now we are able to produce the seed to supply the seed nearly around 3 to 4 million seeds every year. My friend Dr. Kandan is there, and they are happy that that RGC also for the past more than 2 decades they have been producing supplying to the farmers, and consultancy was given by us. Then year-round breeding also we achieved, because a farmer or entrepreneur cannot operate a hatchery for fewer months, because all these fishes are the seasonal breeders. For example, if we take seabas can breed only 4 to 5 months in a season, that is July to October. So once northeast monsoon comes, then they may not be able to produce the seed. So we have developed a customized technology, aquaculture-based technology breeding units, so that maybe 10 months in a year, 10 months except December and January, rest of the months we can produce the seed in the hatchery, so that the hatchery is completely operational, so that the income comes. So it's economically viable. Economically viable hatchery will be available for seed production, that we are able to achieve 2006. Then we have demonstrated from 2006 to even till now, we have been demonstrating nursery farming, then pre-grow, grow, in pond-based system, cage-based system. So we have been demonstrating the farmers, so that the knowledge is transferred to the farmers, so that they can do independently with the scientific practice. They can do scientific practice, so that they can get good revenue. Next slide, please. So these are some of the rearing phases. If you take, there are 4 phases in seabass rearing. One is the fry production in the hatchery, from the spawn, from the egg to, from the mother fish, egg produced, from egg to fry production. So in the hatchery, it is reared up to 25 to 30 days. So if anybody is having the tank facilities, and they can get egg, fertilized eggs from hatcheries, or they can do rearing, within 30 days they can get an income. But for that they have to get facilities like rearing tanks, water intake system. They should have a livelihood culture facilities for algal culture as well as rotifer cultures. Then nursery rearing is another type of rearing facilities where the fingerlings can be produced. The fingerlings are about 3 to 4 inch size can be produced here, up to 10 gram size. From 2 centimeter to 10 centimeter can be produced within a period of about 75 days, 60 to 75 days. So in a hopper, if somebody is having a pond or even open water system is there, the community can come forward and they can get approved from the panchayat. They can do the nursery rearing, and the fingerlings can be produced, and they can supply the fingerlings to the pre-growth farmers. The pre-growth farmers can rear from 10 gram to 50 gram size. This 50 gram size is the size where the growth culture farmers very much require. So the 50 gram size, if it goes, then within 6 to 8 months, the farmer can get definitely 1 kilo size. 1 kilo size can fetch about 450 to 500 rupees per kilo in the farm gate price as on today. So these are some of the different phases of seabass farming. Next slide, please. Seabass nursery rearing, let us see our demonstration. from the various states with the participation of the farmers, we have done it. So this is the nursery. Yeah, next slide, next slide. Yeah. This one you can see, this is an open water system. This is an open water where we have demonstrated in the Maharashtra. You can see Maharashtra district. We have demonstrated. The farmer even, you know, he don't have even his own pond, but if his residence near to his hometown, there is a brackish water body, open water body. So they can do on community basis because these open water bodies can be given with the permission of Panchayat, where as a community activity they can take up. 10 people join together, they can take, or 20 people can join together and take. And this can be, you know, we can promote the self-help groups, especially women's self-help groups, men's self-help groups, youth. We can promote and we can definitely make these activities income generation activity for the farmers. So the technology is available with the SIBA, where a 2 meter square hopper can be installed. A series of hoppers can be installed with the support of the case poles and stocking density and the rate of final numbers per meter cube. We can stock it. And this is a pellet feed. A pellet feed is available. A farm-made nursery pellet feed is available with the SIBA we have developed from our nutrition group we have developed. So with this, because SIBA is the one thing which we have is a predatory phase, a cannibalistic nature is there. So we got to go for regular grading. Every 5 days or every 4 days or weekly twice, we got to go for grading. If you do regular grading, definitely we can get a survivality even up to 70%, 70% we can get. So nursery has to be done as a group, as a team, and it suits more to women's cell groups where they can sit and they can clean and they can grade and they can stock and they can clean the hoppers. All these activities can be taken by the team members and a successful production they can get. Next slide, please. Next slide. Yeah, this is the one trial we have conducted in Nellore district, one of the farmers in Andhra Pradesh. So he is able to get about 65 to 70% survival rate. He is able to get, you can see the series of ponds. So SIBA supplied the nursery seed, 2 centimeters SIBA seed we have supplied, price size seed we have supplied, and they are able to stock it and they are able to rear it. Next slide please. Yeah, this is the freshwater pond. Because SIBA is a univalent fish. It can withstand the salinity from 0 to 35 ppt. Absolutely salinity is not an issue. So even Manargudi, we have through our district, we have demonstrated a farmer from the Manargudi approached us and it's about 2000 meters square pond. In his pond, he has totally spent about 2.8 lakhs and he is able to get net profit of 1.52 lakhs and he with the total revenue of about 4.32 lakhs. So the SIBAs can be taken off with a freshwater pond or even an open brackish water system, and we can do with effective management, with the effective management protocol if we follow, they are able to get a good revenue. Next slide, please. So this is the one we have conducted in Pulikad, Palaverkadu in Tamil Nadu. So we have conducted, there's a woman, it's a tribal women circle groups. So they approached us that they want to take up this particular activity and we have demonstrated, we nearly 2 years we have worked with them and it's a very interesting to see that this particular pond facility is available very close to their residential colony. On morning and evening they are able to spend a half an hour or 1 hour and they are able to get 78% survival rate and they are able to supply their fingerlings immediately to the pre-grown farmers and they get a good revenue. And even now they are practicing and they are approaching us to supply the seed. Next slide, please. So this is the Andhra Pradesh. So Andhra Pradesh is a different kind of farming system, nursery system. In Andhra Pradesh, there is a village called Garladiba. It's near Krishna district. So this village is known as, today known as, it's a nursery hub of SIBAs. In 2005, SIBA has first demonstrated SIBA's nursery in this place. Right now, about 400 acres of farm area presently utilized for SIBA's nursery, and there are about 300 youth involved in nursery and related activities. They not only do the farming, there is a youth involved in seed collection, seed transportation, then seed traders. They supply the seed, they get the seed from the farmers and they supply to another farmer and farm laborers. So it provides employment in this place. It provides employment and they are able to supply the seed. Most of the states, even they are able to supply the seed, even Haryana to the north, Haryana by road. Right? They take by this syntax tank with the oxygen cylinder and all, they take it. Similarly, this down south, even Kerala, they are able to supply. So there are about demand so far here. There are about 25 million seeds. 25 million seeds that are about 2 centimeter size. If we have, then definitely it can absorb such. So that much of farm area is available there, and in a year they can produce about 4 crops. 4 crops in a year they can produce. Because this particular village, Garladiba, in Nellore district, is blessed with Krishna river. Because Krishna, it's located in the Krishna estuarine, that area where a lot of zooplanktons are available. So they collect these zooplankton, naturally available zooplankton, and they regularly stock in the pond, and they get a very good production. Next slide, please. Next slide. Yeah, this is another farmer, Kotishwar Rao. He's also from Garladiba village. And this is actually lease basis. They are taking the farm on lease basis. The lease amount is about 10,000 to 15,000 per acre. And he's having about 11 pond. And you get even a small size seed he wants. 0.8 centimeters. This is about 18 days old. 18 to 20 days old. Yearly fry he takes. And he stock about 15,000 per acre. And he collects naturally available from acetes and crustacean normal forms. And within 75 days, and he is able to get a 4-inch size at the rate of 70% survival rate. You can see the fingerlings and advanced fingerlings the farmer was showing in his hand. Next slide, please. So this is Thrissur district in Kerala. We also demonstrated nursery herring in Kerala, a pond-based activity. In a pond, we have installed a series of hoppers, and the farmer is able to get about 50% survival rate, about 2 to 3 inch size, by feeding with trash fish as well as the later artificial feed. So different states we are demonstrating, as well as we are transferring what are the techniques, what are the methods we follow, because the grading and all those things they go to carry out so that they can get a better survival rate. Next slide, please. So this is another one. Previous slide. Yeah, this is another one. Low salinity Tiruvallur district, Pundi also we have demonstrated. It's mostly nearly around 3 ppt salinity, so almost a freshwater system. A small pond. It is about even 510 meters per pond. We have demonstrated, and the farmer is able to get about 73% survival rate within 21 days. So we have supplied about 6,500 seeds. The farmer is able to get nearly around 5,000 seeds. And of course, the different sizes there, 2.4 to 2 inch, 1 inch to nearly around 2.5 inch size. But we got to go for weekly grading. Regularly, we got to go for grading to get a better survival rate. Next slide, please. This is Navsari, Gujarat. Also, we got demonstrated. You can see a series of farms. Next slide. Next slide. Next slide. You can see the very because morning if we take around 7 o'clock, by 9:30 or 10 o'clock they have to finish the job, so that the temperature before it get hot, they go to finish. So the team, we got to train the team members. So the some people they are good in grading, so they will do very quickly. They segregate the small size, medium size, and big size. They segregate. Some people will remove the hoppers and they transfer the seeds to the bucket. Some people bring the buckets to and supply to the grading people. And grading people again they load the graded size wise graded seed they supply, and they again they go on restocking it. So these are some of the activities, and we need trained manpower to carry out these activities to get a successful survival rate. Next slide, please. So this is some of the economics. If you bar any cultural activity, we go to have economics, because what is the investment cost and what is the revenue. So after deducting the cost of production, what is the next net income? What is the net income a farmer is able to get? So here, if you see, a farmer is able to total expenditure is able to he has to spend nearly around 1,04,500 rupees. And out of which net income is getting a total income of about 2,10,000 rupees, after which the we four lakh rupees. This is the I'm not audible. Hello, can you hear now? Yeah. Next slide, please. Next slide. Yeah, this is the cage culture of sea bass we conducted in Tirshu, in Kodumulu. We have conducted. If you see, so small volume cages. A farmer, if he use small pond, he can have a small volume cages. So each cage is about 2 meters square, and we can get 24 meters square. And he has got about 475 kg, the surveillance of about 18%. In a cage system, you can have nearly about 20 kilo. You can, and the cost of production came nearly about 220 rupees. And revenue is able to get about 1.9 lakh net revenue. Net out of it, he's got about 85,000 rupees. Yeah, this is another one, Maharashtra. Maharashtra, we are able, Maharashtra government is very proactive government, because they asked, they contacted, the minister came to Sibal, instead of looking at a facility. Then after knowing that we have this technology, and they came forward to fund, especially to popularize the cage culture of sea bass in their facility, Sindhudurg district, especially because Sindhudurg district is having a lot of backwaters, brackish water, and mangrove-based backwater is available, and we have demonstrated nursery rearing, pre-growth, growth, culture practices we have demonstrated. Next slide, please. Next slide. Yeah, so you can see that the growth cages, we have tried with the two type of cages, where a frame of the cage with HTB make, or even a GA make. And if you see the to this thing, there is a cell type is there, there is a difference is there. So we conducted on a board, and now we already 2018 we have conducted this for 3 years. Now 2021 we have completed, and we have come out now, and they are doing now independently, and even this particular practice have moved to other districts. Other districts also now they are doing. Thane district, Ratnagiri district also they are now practicing. And Maharashtra government already signed a MOU with us to establish a multi-species hatchery. The sea bass hatchery will be established by us. Crab hatchery will be established by RGCA, and mussel bivalve hatchery will be established in Sevapai. And they already find a site in the Sindhudurg district, and now the paperwork is going on. So very soon we'll be establishing a sea bass hatchery in Maharashtra. And before that, we are already manpower we already trained in Sindhudurg district for nursery rearing, pre-growth, and growth. So how to do the farming and all those things. So when the seed comes, immediately the cultural activities will be taken over in full-fledged way by these people. Next slide, please. So these are some of the the different ways of sea bass farming with the cost effective. What is the material required, and what is the cost, and how we can go about it. Nursery rearing, pre-growth, and growth, everything is there. For example, what is the size, GA pipe, what is the cost, then what is the mesh size, inner mesh, outer mesh size, then what is the approximate cost, stocking density, culture duration, survival rate, and everything we have given here. And this will be very useful for the farmers as well as the entrepreneurs. Next slide, please. Yeah, this is we have conducted in 2019. Our SIBA has conducted a world conference on brackish aquaculture. So we have distributed the revenue about 10 lakh rupees. We have generated in Maharashtra self-help groups. We have our DDG, Dr. J.K. Jana sir, and along with our director, they have distributed our minister also is there, former minister, business minister also is there. The photos, and Mira Kumari madam, our former DDG also is there. So we invited all those farmers of Maharashtra, and we distributed the benefits to the self-help groups. Next please, next slide. Now this is, if you see in one graph, which indicates that what is seabass farming. If you see, almost from the egg to marketable size, it takes almost 1 year. If you have the egg, then to produce the fry size, then nursery phase, then pre-grow and grow, and it takes about 1 year. But every phase there is a revenue, there is an opportunity for a business, because for fry there is a demand, huge demand is there. As on today, for sea bass fry, about 80 to 100 million seeds. If you have the fry, you can easily sell out. Similarly, for pre-growth seeds, they have advanced fingerlings about 5 inch size or 4 inch size. If you have, then you can really save, because nowadays not only for the pond culture but also for the cage culture, especially in Kerala, backwater cages are coming up. And Tamil Nadu, if you go to Ramnad districts, there are a lot of cages, because we have certain issues with our fishermen. Rameshwaram fishermen, they go to other some issues are there, that they have some problem in the fishing because of boundary, international boundary, all those issues are there. So government want to promote alternative livelihood for these fishermen through cage culture. So for the cage culture, cages are available, but what about this stocking material? Stocking material of correct size, appropriate size is very, very important. So these 4 inch, 5 inch size or 6 inch size, about 50 gram, 100 gram size, the correct size. If you are able to supply, definitely the fisherman will get a very good income, and definitely that will the livelihood income generation activity for them. So this is the one particular slide which indicates the entire growth phase of the culture phase of the sea bass. Next slide, please. Next slide. So this is the one harvested chilled live. Even live also now there is a market. You can get a premium price, even 500 rupees, 600 rupees. You can get, and always 1 kilo above is a preferred size. So there is a good, even 500 gram there is a rate, even 750 gram there is a rate, above 1 kilo there is a rate. So any size there is a price is there, and market is there, and life is also there is a premium price market is there for sea bass. Next slide. So this is the one slide. It indicates the entire what is the cost. If you see the egg is around 20 paise we are supplying. Then larvae will be 1, and fry we are supplying about 2 to 20 paise. Single is about 40 rupees, and sea bass around 400 to 500 rupees. Next slide. Next slide. So technology transfer is very, very important, because whatever the technology we develop, it should not stand to the institute or the lab. It should transfer to the beneficiaries, our target clients, so that our technology is transformed into a production model or production mode, and that it become a income generation activity. So in that way, SIBA also assigned number of MOU we have signed with farmers and entrepreneurs. Let us see one by one. Next slide, please. So this is the particular slide shows that they are 3 youth from Karnataka, Gundapur district, Karnataka. After they pass out of BFSC, they approached us that they want to have sea bass rearing, a sea bass farming, a sea bass hatchery. They want to take up, because they told that they don't want to pursue the PG, the post graduation, but they, instead of that they want to become an entrepreneur. They approached us. Then we have given a training, and they came out with the RA-based pickling production unit in Gundapur district. So for that, we have supplied, they invested 50 lakhs rupees. They invested. They get some loan from some source, and they established a small unit by investing 15 lakh rupees. And we have supplied 3 batches of seed. We have supplied, and they are able to get all the 3 batches. They are able to get average of about 70% survival rate. After 30,000 fry, they are able to produce nearly 30,000 fingerlings, and they are able to get a good revenue. Next slide, please. And as on today, now they have signed MOU for the hatchery, and now we already supplied the egg. Last 2 months back also we have supplied the egg, and 1.5 lakh egg we have supplied. Now right now they are having around 1 lakh fingerlings, and now they are doing they are supplying these fingerlings to the cage culture farmer in and around Gundapur. So the cage culture practice, cage culture of sea bass is coming up in Gundapur district, Karnataka, as well as they are sending the ship to even Kerala, as well as into Goa, the west coast. So whatever the demand, it comes from the West Coast agriculture farmers, they are able to meet a small level. So once the hatchery facility is in full operation, definitely not only the West Coast, even they can supply the seed to the even East Coast as well. Next slide, please. So this is another entrepreneur in Karnataka, sorry, in Andhra Pradesh, Kakinada. We have supplied not only sea bass egg, we have supplied even milky seed also we have supplied. So we have supplied about 1 million, about 9.5 lakh sea egg. They are able to sell. They are able to sell about now nearly 1 million. They are able to nearly 1 million seed. They are able to produce both the milky and sea bass. Out of 2.5 million spawn supplied by SIBA. So they got a survival rate about 35 to 40%. They got survival rate. So this is the model. You can just see that they need a live culture facilities, algae, horticulture tanks, and there are some labors are involved in the grading activities. So this model we want to promote in various states so that the farmers long distance to down south to Chennai or even to Sigali to get a seed, but if they take the egg and they can produce the fry or fingerling, and that locally the farmers can get the seed so that the seed transportation cost can be avoided, it will be reduced, that will even cut short their cost of production. Next slide, please. Next slide. Yeah, this is another RAS based model. This previous slide, please. Previous slide. Previous slide. Yeah, this is a RA-based system. This particular facility is available in Cochin, Vipin. So his name is Sadish Kumar, Mr. Sadish Kumar, and he came to us actually to sign the MOU in RA-based system. He want to produce a freshwater system where he is having a land of even he actually in these cases he has taken this land. He has taken this about 10 cents. He has taken on this basis, and now he is having RS-based sea bass farming activities. And now he is having a produce of about 400 to 500 grams, and he is selling 510 rupees per kilo. So he is selling at the rate of 510 rupees per kilo by producing this sea bass with the artificial feeding. The freshwater system is totally land-based activity. Land-based facilities, there is no brackish water, there is no sea water, it's a total freshwater, and it's a RA system. And every day he is supplying about 10 to 15 kilos to the local restaurants. And his intention is that he wants to expand this particular facility in large scale in the coming years. Next slide. So milkfish is also another important species, a herbivore fish. So let us see quickly this what are the achievements we have made. Next slide. So milkfish is a herbivore fish. It can consume even plant-based feed. So the cost of production would be here around less than 100 rupees per kilo. And in a farm gate price, you can get 150 rupees in the East Coast, but in the West Coast, you can get even 200 rupees plus. And this species is actually very good for the polyculture and even biofloc system also. This species is highly suitable. And next slide, please. Next slide. And if you see the timeline of this particular species, yes, 2015 we made a first breakthrough, and we have made a breakthrough in June. Then followed by coming years from up to 2021, continuously we have been producing seed and supplying to the farmers. So by hatchery technology, what we have got something unique from the hatchery technology means these particular seeds are available even wild also. March, April, May, these 3 months you can get milkweed seed from the wild, especially from Pamban Rameshwaram area in the Tamil Nadu, as well as from Andhra Pradesh, Kakinada, Machilipatnam, Krishna district, you can get. Only these 3 months you can get the seed. But by establishing a hatchery, we can produce the milkweed seed even up to October. From February to October, we can produce. Because these are all conditioned fish, these are domesticated. Domesticated almost more than 10 years. And if you do hormonal manipulation, through hormone application, hormone implantation, we can extend the spawning beyond the breeding season. So our ultimate aim is that we want to have year-round breeding of this particular fish throughout the year. Since the cost of production is very less, 100 rupees, even you can get easily 5 tons per hectare, even 50 rupees of profit more than even minimum 50 rupees also. you can get per hectare, you can get 2.5 lakh even 4 to 6 months, because this fish having market, even 250 gram also there is a market. So this particular fish also we have been producing seed and we are supplying the seed to the farmers. Next slide. So these are some of the, in the overall, in a global view, which are the countries they are being farming. Yes, Philippines, this fish is called as a it's a national fish. It's called bangus. Taiwan also very popular fish. Philippines, Indonesia, this fish is very, very popular. Yes, next slide. This particular technology we have established implantation techniques. This we have applied for patent also. We have recently published this paper on aquaculture last issue. Our paper has come. Yes, through hormone implantation, we are able to get a regular maturity and spawning of this particular fish. Next slide, please. This is a photograph actually produced in milk fish seed. Next slide. Yeah, these are some of the demonstration, because we go to popular milk fish is not that popular in some states like West Bengal and all. We go to popularize this fish. So in the number of Deccan Hills, because this is bony fish. This milk fish is having a lot of bones, and especially people from Kerala as well as from West Bengal, they like bony fish because they like to chew the bones and they used to taste it. Similarly, in the northeastern people also they take this bony fish very much. They like it, like freshwater fish. So we would like to popularize this fish in West Bengal. If you popularize, definitely from West Bengal it can go to even northeast, Assam, Manipur, all those states it can go, and we can even extend the market so that there is a whatever the production comes, our Indian market can absorb. So in that way, we have supplied the seed to different states and we have demonstrated. Next slide, please. So this is some of the harvest mela, milk fish harvest mela also we have conducted in different states, West Bengal. Next slide. This is another demonstration and photographs. Everything is coming in newspaper of technology. Next slide. So what is the market trending? If you see from different states, different price is there. Tamil Nadu, Andhra Pradesh not good price because of the boom. The people not prefer this fish. But if you regularly supply this fish, definitely if they taste it regular basis they taste it. Definitely this fish can be demand can increase, and so that the price can also increase, because this fish is not coming regularly to the market. But once people start tasting it, definitely immediately if they taste, then definitely they will like to have, because the taste of this fish is very unique and people should repeatedly taste it. Then only the demand will come. But other states like Kerala and West Bengal and Maharashtra, West Coast, there is a good prices. On Goa, but very good prices. Even it goes up to 200 rupees, 250 rupees also there is a demand for this fish. Next slide, please. This is one Arun Padayar, our knowledge partner. So we have signed MOU to establish a hatchery in Kakinada, and we also signed MOU with another partner, another entrepreneur, RU Swami Hatchery in Raji Hatchery in West Bengal. In West Bengal, there is this place called Diga. They have a lot of seawater resource. So we already signed a MOU and we already supplied the seed. So now 2 years and 1 kilo, 1.5 kilo milk is available. So once the lockdown is over, then we want to go and we want to establish a hatchery. For that, we already signed a MOU. Next slide, please. These are some of the paper cuttings. One when we made a breakthrough in 2015. Yeah, next slide. This is a Atropas rotensis pearl spot. This is a very popular fish in Kerala, Karimi. So yeah, 200 to 300 gram fish can fetch even 450 to 500 rupees per kilo. And this fish is also, we have a customized breeding as well as farming technology. Next slide. Yeah, this fish is having a very unique breeding habit, because a pair, a male and female have to form a pair. A pair can, once a pair is formed, then a regular basis it can spawn in a tank system. So for that we have made a customized RAS tanks, a 1-ton tank, 1,000-liter tank. If you put a pair, then it can spawn regular basis. And if you remove the egg, or fry, then every 21 days it can repeatedly spawn. If you cut it or if you remove the parental care, then again repeatedly it can spawn. So in that way we are able to get a regular breeding, because this particular is a year-round breeding. Most of the months this cichlids it can breed. So we can regular breeding we can get, because the fecundity of this fish is very, very less. As on average, the fish can produce about 1,500 eggs, even though the literature says that it can produce about 2,500 to 3,000 eggs, but as on average, it can produce about 1,500 eggs only. So for mass scale production, we go to have a series of breeding units, a series of tanks, so that regular basis they can produce the eggs. Next slide, please. So this is some of the the breeding frequencies and everything we have worked out. Yeah, in 120 days, yeah, we can get about 8,000 larvae. In 4 months, we can get about 8,000 larvae with a regular with a 17.6. That means almost 18 days interval we can get. This is the data we are get. Next slide, please. Next slide. So this is a breeding unit. You can see the series of tanks has been kept. This connected to a common drainage as well as the inlet canal pipes, and it is separated through the filters. Because sand filter, biological filters, it pass through the same water being maintained. Next slide, please. Next slide. So we also transfer technology to the farmers, in Kerala, and now they are regularly producing a tank-based system, and they are able to get a price of about 1 in size 8 to 10 rupees. The pulse pod seed also can be used as a ornamental purpose also. Not only for food purpose, juveniles ones can be used for the ornamental purpose also. Next slide, please. So this is another farmer in Kerala, in Ernakulam districts, as well as in Alleppey district. The farmers are doing very well, both in breeding as well as in the farming system. You can see a lot of the cages are there. Even cage breeding system also it is coming up in the pond itself. There is a lot of cages, and the seed is produced and the seed collected, then do nursery in the different hoppers. Next slide, please. So this is a self-help group. We have transferred this particular technology to the self-help group, especially in Tirupur, and the several tribe self-help groups. We have provided a facility like you can see the small tanks. We have given 500-liter tanks. We have established for them, and rearing. See, 2 tanks we can use as a breeding unit, or 2 tanks we can use as a larval rearing and nursery production units, and they are able to produce and they are able to sell the seeds. So this is the one model. Even if you have around 1 lakh rupees, if you invest, every month they can get around 10 to 15,000 rupees every monthly basis they can get. So this is a small scale investment, small scale activity. As a part-time activity, per day 1 hour they can spend, the morning 1 hour, evening half an hour. So they can get some kind of part-time revenue. So that is a model we are propagating. Next slide, please. So this is another Alapla district farmer. You can have the harvest he is sowing. He is having a full-fledged facility both for breeding unit, nursery as well as growth. You can see the breeding tanks and nursery hoppers as well as the growing cage culture and pond also is there. Next slide, please. Grey millet wheatle sapple is another important species. Yes, we have worked out this last year. We made a breakthrough and we have supplied the seed to the farmers. And this is also suitable for a polyculture. Especially it can be sold at the rate of 300 to 400 rupees per kilo. In 6 months period, it can go to 500 grams very easily. Next slide, please. So here we have even pond-based breeding unit. We have made a breakthrough last time in Ernakulam, where in a pond there is a goods are available. But this fish, even though it can be formed in the freshwater brackish, but this fish will not mature in the brackish water condition. We need a salinity, good salinity. So we have manipulated salinity using the salt, and we collect the goods from the pond, and here we are able to breed the fish. You can see the RE system, and our former director Dr. Vijayan is there. And we are able to strip. We want to strip this egg and fertilize them, and for larvae purpose, we want to strip this larvae to the hatcheries. Next slide, please. Now this is you can see the different photographs. You can see the larval stages. And right now we have both East Coast stock as well as West Coast stock we are having, and we are able to breed both the stock. Next slide, please. Ornamental is another important area where we are able to work some 5 species, and we are able to. These species are very, very important because this will form as a small scale activity for the farmers. Especially spotted scat, the ornithochromate, moonfish, even crescent crabs as well as Heteroplus. All these species economically important with respect to ornamental. Next slide. The ornamental is a silver moon. You can see each piece about 3-inch piece can be sold about 70 rupees. If you go to Kolattur market, no fish is sold at 50 rupees. Even 1 inch or 2 inch also not less than 50 rupees per piece. So we have developed a breeding technology for this fish, and we also supplied the first batch of seed we have supplied last May. We have supplied to the Kolattur aquarium traders. Now we have been now very good stock. Now we have been regularly we are producing, and our ultimate aim is that this technology will transfer to the ornamental self-help groups so that they can produce on regular basis. Next slide, please. So these are some of the activities. What is the good stock size and what are the breeding methods, hormones, larval breeding protocols, all that we have standardized. Next slide. You can see the different larval stages and growth rate. You can just see. This all we already published in aquaculture research. You can refer it. Next slide. Next slide. Yeah. So we also signed MOU with some of the aquarium fish growers in Kerala, and we have supplied these seeds to them to rear the freshwater, accommodate the freshwater and supply. And definitely once we get some large volume and we get larvae, and we supply to them, and they will rear it and they will sell to the farmers. Aquarium trading. Hobbies. Yeah. Next slide, please. Next slide. Yeah, these are some of the photographs we have taken last year. Yeah, next slide. Yeah, self-help groups. If we supply 1 inch size or even less than 1 inch size, they grow to 2 inch or 3 size and they will supply. Self-help groups, this is Kuvattur. In Kuvattur, there is a backwater, and we have supplied silver moon juveniles, and they reared and they sold to the aquarium traders. Next slide. Yeah, this is spotted scat. Scatterfish is also very, very important. Ornamental species. This fish also, food fish also. Per piece, a small 1 inch size can be sold at the rate of 25 to 30 rupees. Next slide, please. Next slide. Yeah, these are life cycles. You can just see RAS facilities. This technology also we have developed, and we are supplying the juveniles to the farmers, and they rear it and they sell to the aquarium traders. Next slide. Yeah, these are RAS facilities, a small backyard type RAS facility available at Muttukadu station. Next slide. These are all the transfer technology, different farmers, different states. We have given the seed and they are culturing it, and they are these are the capacity building. Once the capacity building develop, then we can give a large quantity and they can put a large volume and they can supply regular basis to the aquarium traders. Next slide. Yeah, these are chromate, another important piece in West Bengal. This is very demanded piece. So we have supplied small one to the West Bengal, and they're able to really piece 40 rupees they're able to sell. Next slide. Next slide. Yeah, this is the same piece in different color. You can see the orange color. Yeah. Next. Yeah, this is another group. Yeah. What are the factors influencing large scale production of sea bass? So right now we don't have many hatcheries, only SIBA as well as RGCA. Only 2 hatcheries are there. So we have to, the more hatcheries have to come from the private players. So once we have already signed MOU, RGCA, SIBA, we have signed MOU with not only private entrepreneurs as well as to the government departments also. We have signed MOU. So hope in a year or two, definitely many hatcheries will be in the pipeline and they will be producing seed, and they can the demand can be met. Then again, if the locally available seeds will be available for the farmers, and stock seed is a criteria, because in the early phase there are a lot of risks there. They want to create the differential growth, cannibalism. So we need trained manpower, and another one is the cost effective feed, because per kilo feed now around 75 rupees. So we want to even bring down the feed, because the sea bass feed is a artificial feed, is a slow sinking feed, and we need to definitely work on the protein requirements. Around 40 to 40% is required. So definitely we got to work out, and financial support should be provided by the line departments for loan, subsidy, insurance, especially to establish a sea bass hatchery, kingfish hatchery, and also the farm facilities. And now the unorganized marketing structure is going on, and we want to really organize so that farmers can get a reasonable price. And of course, the farmer mindset also has to change. that they should not stick on to single species, but they have to go for more diverse species so that they will get every crop they get assured. Next slide, please. Next slide. Yeah, SIPA is working definitely future strategies, our future strategy that capacity building among the farmers, and our ultimate intention is that different hatchery has to come in different states so that the seeds will be available locally, and definitely that will help the mobility of seeds and feeds among the farmers. And the certain activity like milling production can be a small scale activity for the small scale farmers so that it will be a livelihood income activity for them. And definitely more breeding units as well as the group bank we want to develop. For that also we applied for a project from PMSY. We already applied for project for group bank, seed bank as well as the ornamental fish breeding and culture. We have submitted a project, 3 projects. Let us hope that project comes in. Then definitely we will be able to produce a large scale fry and fingerling, as well as we can be able to supply the fertilizer eggs, and we can definitely increase the fish production. Next slide, please. These are some of our publications for extension purpose. Next slide. Next slide. So some of the books also we have published. Next slide. Next slide. Next. Next. This is seed advisories. Yeah, these are the lockdown period. What our activities we have done. Lockdown period also. We never stopped even single day our hatched activity. Whether first lockdown or second lockdown, we have been regularly going. Our hatchery boys, they stay there. Even 15 days, 1 month, they stay there in the hatchery. For them, food, accommodation, everything from the Muttukadu, because live fish has to be maintained with the feeding, water exchange, and everything like our human being. We want to take care of them because every good fish is 10 years old. What we maintain our Muttukadu facilities. So I'm very thankful to my director as well as my officers at higher level for providing all the support. In spite of lockdown, they provided vehicle for us and they supplied labor for us. And each and every hatchery member, they helped us so that even now we have our own operation. We are able to supply these seeds regular basis. And we are even, you can see Karnataka, we have supplied the egg, and you can see the transportation even Karnataka, because seed transport is a permission. So that we are able to. Next slide, please. Next slide. This is some of the harvest. We have harvested the fish from West Bengal as well as from the Navsari Research Center, Gujarat, and we have supplied in lockdown period. There is no fishing activity, so on a small reasonable price, we are able to supply the fish to the local public and in a reasonable cost. We are able to supply. Next slide. So this is the last slide. Before I'm going to thank, let me tell you about this particular slide. You can this particular slide showing your father, his children, his father, that means grandfather, father as well as children. So 3 generations are there. So this is the message we would like to pass on today to our younger generation, that fish farming, aquaculture activity, this is his own farm. He is having about 2-acre farm, and this pond is produced the sea, and he stock sea bass, and he stock pearl spot and some other species, and he gets some revenue from this pond, and he get regular income. And the message is here that aquaculture activity is passed on to it now never stopped with the present generation, and it is passed on to the young mind, the children. And definitely this value of the fish culture, the value of the healthy food fish, it can be it can be transformed to our younger generation, so that aquaculture will definitely be taken up a large scale, and it can meet the food demand in the future. Thank you very much, sir. Thank you.
Thank you, sir, for the wonderful lecture. It was really informative. Now I request the participants to post the questions in the chat box. Sir, there is one question from Sangeeta.
Yeah.
Suppose we have land area, and if we want to set up a hatchery, will they help us in setting up? If so, what is the procedure or cost?
Yeah, if you should have a land in the coastal area. The seafront beach should be there should be direct connection to the sea. We should have that approach road. This should not be in between this many private land or even forest land. So if you have even 2-acre land, yes, we can definitely, you can write a letter to us, and definitely you can sign MOU with us, because we have a ITMU unit, that is a business development team is available in our SIBA. So you can come to them and you can discuss with them. And first we sign MOU, then we can in a phased manner we can take up activity, and within 1 year we can have a full-fledged hatchery by one-to-one discussion, and definitely 5 million seed capacity production. We can get nearly around 3 crores. Roughly we estimate around 3 crore rupees. It estimates. Then definitely you can get a regular production. Because the demand is, for seed demand, I already told you, there are about 80 to 100 million seeds per seed, but there is a huge demand, and definitely even 5 million is nothing. So definitely you can sell each seed, you can sell nearly around 3 rupees per seed. So net profit, even you can get out of this net profit. All that we can work out through our MOU. Yeah.
Next question.
Sir, myself principal, DG courses Bhupati Bhaskaran. My point is the bioflog method, which we are discussing now, and which is spoken a lot throughout India. Now, what do you think about that? Some say it's a failure, some say it is a success. Some say the alkalinity and the other nutrition values have to be balanced and not overdosed with nutritions and so on. What is your opinion, sir?
So bioflog, actually it is applicable only hybrid species like shrimp, not for the fish. Maybe freshwater system, they may be doing it with this catfish and other tilapia and other species. But as far as brackish water is concerned, this bioflog model is already going on. In a land-based tank system, people are doing it. We have one Dr. Akshay Panigragi, he is our expert in our institute. Maybe he can provide, because he has done a lot of research on that, but it is successful one provided you to follow the scientific methods. And there is a proper stocking density is there, and what is the material to to produce a bioflog, what is the source, what is the carbon source, all that there is a this protocol is there, and all that we have to follow means economically viable production we can make it. If unscientific way, definitely if you do mean, then there will be a problem, because there is a aeration, continuous aeration is required, then there is a biomass is there, then the unutilized only it is whatever the waste comes is transferred as a used as a food. So these are all chain of activities are there. It's a very dynamic system. Bioflux is a very dynamic system, and we have to monitor all the nutrient value and everything, and input everything. So it is actually it's better you can discuss with our Dr. K. Panigragi. So we can give the detail, but it can be done definitely, provided it should be scientifically followed.
Okay, so my second question is, with regards to, combined cultivation with paddy fields, sir. Because paddy field, with 2.5 months, we have water, water retention. And only in the last 15, 20 days, we just drain out the water and we dry the land and we start cutting the crop. So if we go for long-term crops, nowadays, the 6 months crops are also come up. So in that way, will it be possible to advantages to have double system, like fish farming as well as, in a very less 2 inches of water or maybe 3 inches of water with the paddy cultivation?
So this, which state this saline which state is a freshwater system or is a brackish? The paddy field.
Paddy field is freshwater, sir. Freshwater.
Which district? Can I know which district it is?
This is Tiruvallur district. Tiruvallur district, sir.
Tiruvallur district. Okay. It's a freshwater system.
Yeah.
It's a freshwater system.
Yes, sir. Freshwater system.
In a freshwater system, yes. Usually, what they do in Kerala and now only the Pokali, they do 6 months fish and 6 months they go for a Pokali, right? Paddy cultivation. That's what they do. So here, if you want both together, if you want to do together shrimp, together fish as well as paddy, then definitely you go for species selection. So species selection, if you want to 2 inch, 3 water, you cannot do anything, because even nursery rearing, you need at least 1.5 feet water depth. Even nursery rearing also can be done, provided if you have water depth around 1.5 feet. A small scale nursery rearing, for example, even any freshwater fish, you can do it. But if water depth is more than 1 meter is available, yes, you can go for any kind of fish you can go for.
So the water depth comes as a main criteria here in cultivation. Is that right, sir? What you can understand?
Yeah.
Okay. Thank you, sir. Thank you.
Thank you. Thank you.
I'll collect more information from our organizers of this program, and I will definitely approach you in various other thoughts, processing thoughts also, sir.
You are most welcome, sir. Definitely you can contact, sir. We are here
Thanks.
to help you. Definitely.
Thank you, sir. I was really taken up by seeing your slides, where 65% of it is inland and only 35% is only out at sea. The fishing market is. That is something great that inland is developed a lot in
Yes,
meeting
yes,
the needs. Now, I hope even this 35% also is taken care of, because you very well know Tamil Nadu coast is now becoming very, very much difficult to do the fishing in deep waters. Because of various other encroachments, Sri Lankan borders and all these things which are coming up, and which is threatening everybody, particularly down south, Kanyakumari and other places. This inland fishing developments will help them a lot to have a new life, rather than because they know only about fish and nothing but fish, so they can just switch over faster and they can adapt to the new systems.
correct. Yeah.
Thank you, sir.
Yeah.
Thank you.
Thank you. Thank you.
Thank you, sir. Due to the time constraint, we will forward the questions to the speaker. We will share the email ID of the speaker. The participant can reach out through the mail. Thank you, sir.
Thank you, Dr. Kailasam, sir. Thank you so much.
Thank you, sir. Thank you, sir. Thank you.
Thank you, Vilasam sir. It's very fortunate to have your lecture.
Thank you. Thank
So,
you.
so now you can proceed with the
Okay,
next
good morning, Kailasam.
And
Sir, good morning, sir.
Thank
Good morning,
you.
everyone, sir.
Thank you for your talk.
then move. Okay, sir. Moving on to the next session, the next speaker is Dr. A.S. Sahul Hamid, Coordinator, Aquatic Animal Health Laboratory from C. Abu Hakim College. He is OIE expert for white-tail disease of prawn in World Organization for Animal Health. He is the chairperson of International Committee on Taxonomy of Viruses. He is also a research consultant in Kuwait Institute for Research. He is also a member in scientific and technical appraisal and advisory groups on agriculture, animal sciences, and allied science, DBT Government of India. Sir is having 140 research publications in reputed journal with cumulative impact factor 250, citations 4,458, and H-index 40. He has handled 30 major research projects from major funding agencies. He has developed 4 products for farmers, and now it is commercially available in the market, and the brand name is Dr. Sahul's Kit to detect the virus in the shrimp, and Immuzone to control the white spot syndrome virus in the shrimp. During his research period, his research contribution played an important role to enhance the research and development in our nation. With this short introduction, now I would like to invite Dr. Sahul Hamid, sir, to deliver the lecture. I welcome you, sir. Over to you.
Okay, thank you. Thank you for the nice introduction, and good morning to everybody. So I can share the slide now.
Yes, sir.
Sure, sir.
Please, you can share this. Sir.
So 1 minute. I have to go to settings. Share. Please share from the slide sharing. Listen now. Listen now. Your screen. Okay. That is not.
Wait.
This is your entire screen. Hey, 1 minute. Okay, my Now it has come now?
Yes, yes.
Okay, very good. Okay. So good morning to everybody. So respected my teacher, Dr. Altaf sir, and the registrar and organizing committees, Sendil Nathan and other friends. So my dear participants, good morning to everybody. So first of all, I thank all the organizing committee members and my teacher also. So today I'm going to talk on the viral disease management using biotechnological tools in shrimp culture with reference to WSSV. So why I have taken the WSSV here is it is one of the very important shrimp viral pathogens, which cause severe economic loss in the country, not only in our India, throughout the world. It is just like a pandemic, like Corona. So just tell about our laboratory and what we do here in the 2 or 3 slides, then I'll go to the subject. So our laboratory is Aquatic Animal Health Laboratory. So we have separate website, so you can visit our lab through our website. So it is a part of the Abdul Hakim College. So our lab has been accredited by the NABL. So that is our lab is located near Vellore, that is in the Chennai-Bangalore highway, just 120 kilometers from the Chennai. So our laboratory has been recognized by the World Organization for Animal Health. It is called OE, it's a OE reference laboratory for vital disease. So this is the only laboratory in India having this recognition. So what we do in this laboratory, because we have so much of fund, everything, then the people should know what we are doing here. So actually our main research work is being carried out in our laboratory is disease of aquatic animal. So we work mainly all the viral pathogens, bacterial pathogens, and fungal pathogens. That's called white spot syndrome virus, IHNV, IMNV, HPV, MRNV, fish nodovirus, tilapia lake virus, cyprian herpes virus, EHP, it's a fungal parasite, then bacterial pathogens. So in this, what we do here, that is we work for the developing the diagnostic tool for all the pathogens. Then vaccine development. So we developed the vaccine for the world virus vaccine, just like Covaxin. So recombinant and as well as DNA vaccine for the fish. Then immunostimulant we use for herbal immunostimulant as well as viral immunostimulant for the shrimp and prawn. So we also work on that production of the double-strand RNA. Already we standardized, so we already completed the field trial also. So we developed the double-strand RNA specific to WSSV to control the viral infection in the farm. So we also developed the nanoparticles for delivery of the vaccines and double-strand RNA in the farm conditions. So in addition to that, we also have that one surveillance program funded by the ICR NFDB to monitor all the diseases in different districts of the Tamil Nadu, Andhra Pradesh, Assam, and Jammu Kashmir. So in addition to that, we also work on the fish cell line for development as well as application of the fish cell lines. So what is the outcome of our research work? So actually that is this is our real outcome of this all the R&D projects. So we have published more than 140 international publications. So more than 400 citations have been cited for our publication by different authors. So our H index is 40, then I index is 78. So not only that we do the research and develop the product, then we also develop the manpower. So far we produced 24 PhD candidates, and also we trained more than 150 trainees in the PG scholar, PhD students, everything for in molecular diagnostic techniques as well as cell culture techniques. So the products what we developed from our research work is the PCR kit for the shrimp viruses and prawn virus and fish virus, and also we have the immunodiagnostic, it's called lateral flow immunoassay. So then we also develop that develop the products, that's the ImZone. So these are all products have been commercialized. So in addition to that, we also do the service for various national and international organizations. So we have established 2 repositories with the funding of the DBT. So one repository for fish cell line repository, other one repository is viral repository for viral pathogens of aquatic animals. So now we'll come to the subject now. So already that our Dr. Hailasan explained about the importance of the brackish water as well as inland fish culture. So aquaculture is a multi-billion-dollar industry because it provides protein-rich food as well as employment and revenue generation. We are exporting more than 50,000 crore. So mainly that in the export main component is a shrimp only. So if you go for culture in large scale, so the problem is a disease. So disease are now the major constraint and limiting the aquaculture production. So disease of viral and bacterial are very, very important because now we will come to know that the viral disease is very common now. So even common know about what is virus now because of the coronavirus. So the loss estimated due to these diseases is more than $10 billion globally, then $6 billion in Asian countries. So the mainly loss due to the WSSV only. So that is the disease of So actually more than 20 viral pathogens have been reported in the stream, but only some viral pathogens are very important and responsible for severe economic loss. So what are the pathogens encountered in the Indian ecosystem is, so these are the viral pathogens encountered in our Indian ecosystem, particularly shrimp culture. So these are the WSSV, IMNV, IHNV, HPV, and NBV. These are the viral pathogens of the shrimp which affect the shrimp. So among these viral pathogens, the three are very important. One minute, sir. Okay. So that white spot syndrome virus, IMNV, IHNV are very important and responsible for severe economic loss in shrimp. So that another important pathogens for freshwater prawns, because MRNV, the macrobergin, rosebergin nodovirus, and the extra small virus particles. So these are a very, very important pathogen. Even once upon a time, our freshwater production was more than 40,000 ton. But because of this virus, that is now totally collapsed this industry. Now the production is only for 2,000 ton now. So then another important pathogens are the viral pathogens of the fish nodovirus, one of the very, very important virus. It causes severe mortality in the young ones of the sea bass. So then next to the tilapia leg virus, it's another important virus. It affects the tilapia culture. Then cyprian herpes virus, it affects the carp and carpedema virus. These are the important viral pathogens encountered in Indian system. So in addition to that, we have the bacterial pathogens, the important pathogens. Then another important parasite is endocytosis hepatopene. It's one of the very, very important microsporin parasite it's a fungal parasite. So it causes severe, not the mortality, it's severe economic loss due to the slow growth in Vaname. Okay, so any pathologist they'll present this important slide here. So I have modified this slide slightly to make the people to understand more on this development of the disease in the system. So three very important factors which are responsible for development of the disease in the system. So one is a pathogen. Pathogen is an important factor, then host and environment. So these three important factors interact with each other and develop the disease. So what is the role of the pathogen? So the pathogen enters into the system, it causes the disease. So what is the possibility of the entry of the pathogens in the shrimp culture? So suppose that your brooder or your seeds to be stocked are not properly screened for the viral pathogens, the pathogen enters and cause the disease. Suppose if you are not prepared the pond properly, not eradicating the carriers, reservoirs and other hosts, then pathogen enters. In addition to that, if you have not prepared the pond properly, so there is called persistence of the pathogen in the pond. That will be very, very important. For example, WSSC survives 12 days in seawater, 19 days in sediment after drying also. Even suppose the pond is not drainable, the virus will survive more than 45 days. Suppose if you not properly prepare the pond, suppose that if you stock new stock, then the virus will be persist in the pond bottom, then cause the disease. You lose the stock. Then another important thing is failed in monitoring of the pathogen. So that's very, very important. Once this seed is stocked in the farm, then you have to monitor. So if we don't monitor properly, again the problem of the pathogen entry into the system and cause the disease. So sometimes the live feed also act as a carrier for the disease that carries the pathogen into the system. So water source is very, very important. So if the water is not properly treated, the pathogen enter into the system. So these are the possibility of the pathogen enter into the system and cause the disease. Then what about the host? Host is also another important thing. Suppose overstocking of the shrimp high, there will be high biomass. So the high biomass make the animal to susceptible to the disease, then the animal get the disease and cause the losses. Handling. So we should not handle the animal frequently. Suppose handling of the animals also making stress and make the animal to susceptible to the disease. Then underfeeding as overfeeding is a problem. Then nutritional deficiency, weakness of the shrimp, then poor quality of the seed and feed and cannibalism. So these are the very important, these are the factors which make the animal to susceptible to the disease. So then what about next to the environment? So the environment should be proper. Suppose the extreme temperature, extreme pH, low dissolved oxygen, abrupt changes in the salinity, gas supersaturation, poor pond preparation and algal bloom, ammonia, nitrate levels, lack of fertilizer, clear water. These are the things make the environment bad and leads to the development of the disease in the system. Yeah. So now that is just we are coming to the subject now, white spot syndrome virus. So I already explained why we have taken the white spot syndrome virus, because it's very, very important pathogens. So that's why we have taken this, this subject, this lecture for how to manage the viral disease. So that research on our WSC in our laboratory has been initiated in 1996. So we have isolated the WSC for the first time in India from the pond period infected fish. So we published the article in 1998. So actually, this is one of the very important pathogen, the most threatening viral pathogen in the system, not only in India, in all shrimp-growing countries, and responsible for huge economic loss. Actually, in India alone, that SIBA has estimated about that the loss of the shrimp due to the white spot has estimated about more than 1,200, 2,000 crores. So it's not only confined to only 1 year, that's every year. So even that this year also we have lost shrimp due to this viral disease. So that actually this virus has been reported for the first in Taiwan in 1992. So it was we observed that white spot syndrome virus infected animals were in 1993 in India. So that between that 1993 and 2000, it spread to almost all the shrimp-growing countries. What is the clinical sign of the disease? So the clinical signs develops at different stages. So the first initial stage is the animals become lethargic and stop feeding. So later stage that we can see the appearance of the white spot. You can see the white spot on the suprathoracic region, even sometimes abdominal segment. Even you can, in the severe stage, you can see even the distal abdominal segment. That's called sixth segment also. This is a white spot. So actually that is after getting this infection, just before dying, this animal swims on the surface of the water before dying. So that I will explain why this animal dies before, swims very fast on the surface of the water before dying. So that actually in histopathology, there is infection characterized by the cells have the hypertrophic nuclei containing the intranuclear inclusion bodies. That's one of the very important characteristics of the white spot syndrome infection. Actually, we have isolated the virus. Actually, it's very, very laborious and it's very time consuming to isolate the virus from the infected animal. So we have standardized this, this virus has been purified by ultracentrifugation, the supersensitive gradient. So then we also studied the morphology of the virus, as well as external morphology, as well as internal morphology as we studied. So the intact virus, actually this is the largest animal virus. So its intact viral particle is elliptical shape, measured about 266 nanometer in length and 112 nanometer in width. So actually we also removed the envelope. Actually it's envelope virus. It's a DNA virus, it's envelope virus. So we have removed the envelope by treating with the Titanic 100. So we have seen that nucleocapsid. So the nucleocapsid is just like bullet-shaped structure. The length of this viral nucleocapsid is 420 nanometer in length and 68 nanometer in width. So it has a cross striation, and one end is blunt, one end is pointed. So actually, this is highly pathogenic virus. Actually, that even in the farm, that mortality occurs within 3 to 5 days. So entire mortality will be 100%. So entire farm will be infected with this one within short period. Even we have in our laboratory, so we observed that this virus caused mortality just in 48 hours by intramuscular injection and 5 days by oral feeding. So in the Pinus Marodon, in Pinus indicus also we observed same kind of result we got. So it's highly pathogenic. So we also actually for any viral infection, so there, so actually we have studied so many viral pathogens of the shrimp as well as fish in our laboratory. So once you isolated the virus, you can study the pathogenicity, then we have to go further to study the tissue trophism of the viral particles. So the tissue tropism will be very useful to select the tissue for diagnostic purpose. Even for example, coronavirus, we are taking the swab from the nose as well, nostril from the mouth, because there is a aggregation of the viral particles in the particular area. So like that, each virus having the characteristics of the accumulating the viral particle in particular organ. This is called susceptible organ to the viral particles. So like that, we have studied the tissue trophism of the WSSV in the shrimp. So we have used a different methodology to study this trophism, PCR, Western blot, biopsy, histology. So we observed that, so all the results revealed that this virus distributes, that is found in almost all the mesodermal and ectodermal origin organs. So they're all the mesodermal, vital, these are all the vital organs. It aggregates almost all the organs. So we have to select any one of these organs for diagnostic purpose. You can see the PCR result. You can see the band in almost all the mesodermal, ectodermal origin organ. This is Western blot. So we also seen that pathogenicity. We have taken the viral inoculum prepared from the different organs. We found that is a hemolymph caused 100% mortality within 48 hours. That means that the viral is more aggregated in the hemolymph. So like that, almost all the organs are except hepatopancreas. The hepatopancreas is endodermal organ. So this virus cannot replicate in the endodermal origin organ. Even histopathology, this is I told in my previous slide. So there is this one of the very important characteristics of the histopathological examination is the hypertrophic nucleus. This is hypertrophic nucleus. Inside you can see the intranuclear inclusion bodies. So then we also studied the biochemical, physiological, and hematological changes in the infected animal. So this is one of the very important findings. So we found that what is the reason for the death of the WSSV infected shrimp. So even for example, coronavirus patient, the patient dies because of the hypoxia. See, they're not able to consume the oxygen. So here also like that. Actually, that is, we have seen that there is a significant reduction in the total hemocyte count in the infected animal. So actually, another important thing actually is the hemolymph, that's called body fluid of the infected animal, failed to clot. The clotting mechanism failed. So there is a reduction in oxygen consumption and ammonia excretion. So that respiratory system failed now. So that is a disinfection of the respiratory system and causing hypoxia in the tissue in the infected animal. So the infected animals die due to anoxia. So that even that animal, plenty of oxygen available in the medium. So the infected shrimp cannot take the oxygen from the medium because of the damage of the gills caused by the virus. Same in coronavirus also, that the lungs will be totally damaged because of the viral infection. The patient not able to get the oxygen. So same kind of here also. So the WSSV affected the gill and damaged the total gill, and the animal dies because of the anoxia, not able to take the thing. In addition to that, so this virus interferes with all the vital function of the animal. So that is the reason the animals dies in short period, like 48 hours after getting infection. So then actually this virus is very, very difficult to eradicate in the system. So that is the reason every year we are getting this infection in the farm. So why it's very difficult to get? Because it has a very wide host range. So this virus, actually some of the virus have the restricted with particular host, but here it's not like that. So the host range is crab, copepods and other arthropods, sometimes insect also sometimes here. So even we have studied that host range in our laboratory. So we found that more than 36 hosts for this virus. So all the peanut shrimps are susceptible host. Then Macrobrachium rosenbergi, Edella, lamprey, and 22 lobsters and 20 marine crabs and 2 freshwater crabs. So these are the hosts susceptible to this virus in our studies. In addition to that, other people also studied that. They found that nearly 100 species of arthropods have been reported as a host or carrier for this virus. So that is the reason it's very difficult to eradicate the virus completely in the system. So there are different molecular detection tools are available for the WSSV detection. So this helps the industry to at least to prevent the infection in the system. So we also developed the diagnostic tool. So there are single step PCR available, nested PCR available, semi-nested, real-time PCR available. Even the people know that real-time PCR now only after the emerging of the coronavirus. Even that our aquaculture people, they know all the PCR, RT-PCR, real-time PCR just 15 years, 20 years back. So we adopted this PCR technique in the 20 years back in our aquaculture system. So we are very familiar with all the kind of diagnostic tools, even including farmers also. So actually we developed a kit for to detect the shrimp viruses. So we have commercialized in 2006 that PCR kit in the name of the Dr. Salt's PCR kit. Actually we have transferred the technology to one company called MARINE Biotech. So then they want to commercialize this product. Then okay, we accepted. We have made a MOU as well as technology transfer. So we want to that CEO of the company told me that Saul, we are going to give the name. That's household for the kit. Then I was very happy. Okay, you give my name for the kit. Then immediately he said that no, not only that like that. Actually, you have a very good name in the industry. Suppose if the kit works very well, then your name will be there. Suppose if the kit doesn't work, then your name will be collapsed. But hope that now the kit is working very well. Still, I have the name in the industry. So that what is the salient features of the kit when compared to the export kit, import kit? So actually it is more sensitive and specific to the WSSV. It has a very short cycling, PCR cycling parameters. So within 4 to 5 hours, you can finish the entire reaction. So actually this is very based on the dominant protein of the WSSV. So you cannot miss any, it won't give you any false positive or false negative results because of we have designed the primer to target the conserved region of the WSSV. So it is a semi-quality 2 steps and user-friendly protocol. So master mix is given separately in the PCR tubes to avoid the contamination. Even the import kit, they'll give the master mix at only one volume, just like for 10 ml or 15 ml. But we are giving here, separately we'll give them all the PCR tubes. So if one tube is contaminated, that particular tube only they discard. The other tube can be used. So we have given the positive and negative controls given separately. So detect the WSSV within 1 hour of post-infection. So suppose, for example, the virus entered into the animal just 1 hour before. So that can be detected by using our kit. So it's a very fast and accurate DNA extraction protocol we have. So this protocol is very useful for other assay also, like ESP and IHLV. So it's very, very cheap and reliable. So actually we fixed the rate 10,000 in 2006. Still we are selling the same 10,000 for this one kit. It is very useful to screen the brooders, PL and pond cleared shrimp. So actually what is the use of the PCR shrimp culture? To screen the post storage for the WSSV before stocking, to monitor the stock in growth ponds, to study the expression of the immunogene, to understand the status of the immunity of the shrimp. So this will be very useful for all these activities. Actually, so we also have the real-time PCR for to detect the virus as well as to immunogenes expression. So we use here cybergreen as well as for Tagman probe also, but I don't want to go for the technical things here. So actually, we mainly will work for the benefit of the farmers only. So actually, we also developed the technique of the immunodiagnostic for the white spots in the virus. So just for we have developed the anti-serum. So then actually previously we used the viral protein purified in the pure viruses. It is a very laborious technique. So suppose we get just for 100 ml of antibody, so we have to work for months together to develop to get the pure viral protein. So we have to propagate the virus and isolate the virus and purify the protein and immunizing the animal with the purified protein. So it's laborious and also very time consuming. So then what we thought is we cloned the VP28 gene. So instead of going for traditional method, so then we have come to we applied our recombinant DNA technology. So actually we have amplified the gene by using the gene specific primer, the VP28 gene. Then VP28 gene we have cloned in the cloning vector, expression vector, PSB. Then we have transformed into E. coli. Then again we have transformed into the expression host. Then we produce the expression in large quantity. Now we are able to produce that VP28 protein, recombinant protein in very large scale, even in gram we can produce. So this protein we use for to developing the antibodies, as well as even we use this recombinant VP28 protein as a immunostimulant to induce immunity of the animal against the viral pathogens. So this is our purified protein. Then we raised, we are confirmed with Western blot. So we also studied with different organs of the infected animal. So our antibody is very efficient to detect the disease virus. Then we developed that lateral flow immunoassay. So it's very simple method. So even that the farmers can do himself. Within 10 minutes we can get the result. But I cannot say it is highly sensitive like PCR. But the preliminary screen can be done by using this. Even we recommend for coronavirus also for this kind of screening. So it's called antigen screening. So within 10 minutes we can do that. We can finish off that. Even anybody can do this. So the principle is, so actually the antibody, we have conjugated the antibody with the nanoparticles. So we have load that conjugated fluid nanoparticle antibodies here. This is the antibody specific to the BP20 protein of the WSSV. So we also make one lane here. This is also antibody specific to the white spot center virus. It is a control line. We have the anti-IgG antibody. Suppose we put the sample here containing the virus. So this sample will flow like this. So the viral antigen will bind with here. Then all the viral the antigen antibody complex will flow. Then here that already the antibody is there, it will assemble here and develop a color. So just you can see here. So this is the negative. This is the kit using the negative sample. It's a positive sample. So the negative sample, you have the band. It's called test line. Suppose this indicates that the kit is working. So if we get the two band like this, the sample is positive. So our antibody is highly specific to the WSSV. Even the antibody used to react with the other viral pathogens, so we have not got any reaction. So it means that this antibody is not cross-reacting with any of the other viruses except the WSSV. So we have also compared the sensitivity of our lateral flow immunotherapy with the PCR as well as real-time PCR. So this kit can detect the WSSV infected animal containing the 240 nanogram of the total protein. So it also detects the WSSV in the sample containing more than 100 viral particles. Suppose the sample contains more than 100 viral particles, so this kit can detect the virus in the sample. So it's also very useful for early detection. So then, okay, then here you may ask one question here. So when the sensitive diagnostic tools are available in the market as well as in the system, then why you want to go for lateral flow immunoassay? It is less sensitive. Actually, the PCR, this PCR, it costs about 1,000 to 1,500 per test. Then the reverse transcripted PCR to detect the RNA virus cost about 2,000 to 3,000. So we have to depend upon the technical person. So we don't know whether the technician is doing or not properly because the sample is our sample. So that is in that case, then this kit will solve the problem. Why we are interested to develop this kit? So actually in the system, that's the aquaculture system, so the farmers take the seed from the hatchery and go to the PCR diagnostic lab. So the problem is here is, it's just like a medical field. Here also the hatchery people and lab people, they have some connections. So even though the sample contains the positive, the people that give the results negative. So the farmers are affected. So in that case, even the farmer also very intelligent. Instead of giving the sample to one lab, so give sometimes they give the sample to the two labs. So they are giving the result properly or not. So even sometimes that hatchery people sometimes will add that EDTA into the water while giving the seed to the farmer. So the EDTA inhibits the PCR reaction. So there are so many problems in the PCR. Even you may be knowing that the problems we have in the coronavirus in the, detection by real-time PCR. So in that case, this kit will solve the problem. So even the farmer can take this kit to the hatchery, and we can go to the hatchery and take the sample and we can do it, analyze there itself. If it is a negative, it can go for the negative, it can go for the PCR for further confirmation. If it is a positive, he can discard, he can omit, you can deny the taking the sample, you can go for next hatchery for seed. In that case, what will happen is, so this is a user-friendly kit. The cost about is only 200. Suppose the farmer can save 800 rupees if the sample is negative. Suppose if the sample is negative, he can spend 200 rupees extra for going for the PCR for the second confirmation. So in that case, the farmers can save the animal, save the money, as well as the result is very highly confident. as well as no need to depend upon anybody. So that is the reason we developed this kit. So we have transferred this technology to the one triple based company. So we are going for large scale production and for commercialization. So this technology has been transferred to this pathogen technology in the presence of our DBT advisor, Dr. Rawat. Okay, now we'll come to another aspect. That's actually that is our in the morning presentation. Our Kailasam told we should not depend upon. We have very vast aquaculture resource, so we should not depend upon the single species. So once you depend upon single species, if anything happens, this entire system will be collapsed. This happened in 2010, not in 2000, even 2000 itself. So the entire system was depending upon only one species called Penis morodon. So because of the white spot center virus, the entire system was collapsed. To make use of the same resources, the government of India introduced the Penis vaname. So in 2010, it was introduced. There was a huge production. There was no mortality. Every farmers enjoyed with huge production of the vaname. So everybody enjoyed. Everybody earned a lot of money. Then again, the mortality started in 2014 to 15. So everywhere that is everywhere the people are crying. So we have the problem. Everywhere the huge mortality in the Penis vaname. So the system is only a white patch in the abdominal segment, then immediate mortality. So one meeting was conducted by the SAP, Society of Aquaculture Professionals. So there was a hard discussion was going on there between the scientists and as well as entrepreneurs. So that time I also attended the same meeting. Then I requested all the entrepreneurs to assemble after the meeting. Then we discussed with only few people, even including our scientists of the SIBA. So then I asked if anything happens next time, if I find if I encounter same kind of problem, immediately inform. Then I will come and collect the sample. We do that analysis. Then after the meeting, just in 1 or 2 days, immediately I got the call from one farm from Nellore. That is the farm is located near in Ramatirtham, near in Nellore district in Andhra Pradesh. Then immediately I went there with my team. That is in 5th May 2015. So we have seen that is more than 50 farms. That is a very big farm. So more than they have 100 ponds. So every there is a animal died. Every hour there's more than 50, 100 cases of animal dying every hour. I was there for 3 or 5 hours. So even I could see that more than 2 or 3 tons of dead animals. So there is severe mortality. Then we collected some of the moribund as well as dying state animal. So then we brought to the laboratory. So then we screened that infected animal by PCR and RT-PCR using the private specific to all the known and DNA and RNA viruses. Sorry. So then the samples were negative to all the viruses. Then we don't know. So actually that we have all the primers for almost reported virus, non-reported virus of the same in our laboratory. Then repeatedly we have done 2 or 3 times. We have analyzed. Then every time we got the negative. Even scholars also for doing independently. Every time we got negative. Then immediately we conducted the infected experiment. We prepared the inoculum from the infected animal. So we use 2 types of inoculum, with filter, without filter. Filtering with 0.2 micron, a membrane filter, and without filter. So both inoculum caused 100% mortality. That means then again that we have analyzed the experimental infected animal by same, that PCR and RT-PCR for all known viruses. Then again we got to the negative. Then we didn't know that what to do further. So then we came to know that there is some viral pathogen, because that inoculum filtered through 0.2 micron filter caused 100% mortality. It means that there is no bacteria. This ruled out. So now only the viral pathogen is responsible for this mortality. Then we conducted a mini experiment related to the white spot center virus infection. Everything positive, but getting negative by PCR. So the only that we got the thing on experiment is actually the white spot center virus causing mortality on the freshwater crab. That's called Paratilphus hybridomus. So I asked my student, okay, just only one experiment we'll do finally. So you inject the viral inoculum to the freshwater crab, then we'll come to know what is there. We injected the freshwater crab, again, 100% mortality in freshwater crab. Then we thought that definitely it is due to the white spot only. Then we decided to use the primer specific to VP28 gene. I already told, so our kit is having the primer for VP28 gene. So VP28 is a highly conserved region, that is. Then we got the positive. So then actually that primer we used that specific to YRF19 in that experiment. So that shows negative. Then we got the positive for our VP28. So now we got the two strain in our lab now. That is the old strain. Old strain we got the positive for both YRF19 and VP28. The new strain that is called isolated from the pinnace 1MA, we got negative to the ORF19 and positive to VP28. Then what we designed is, we designed a primer on either side of the ORF19. So what will happen, whether anything's happening? Again, we got negative. Then from that, we came to know that there is a deletion in the genome of the new strain of the WSSV. Then we thought some mutation occurred. Then actually, then we have sent the two strain, old strain as well new strain to IGIB. That's called Institute of Genomics and Integrated Biology for sequencing. So we have whole genome sequencing we have done for the first time in India for both strain, old strain and new strain. Then this is we have used actually that genome sequencing for the shrimp virus is very, very difficult, because that is when you go for purification of the virus as well as viral DNA, so always there is a contamination will be there for host DNA. So for that we use the whole genome sequencing. There is a new methodology they found for use this methodology. So we have whole genome sequencing done for both strain. Then we found that there is a deletion of 13,170 base pair in the new strain of the WSV. So that also in 5 regions. The first region, there is a deletion of 198 base pair in YRF 14, then deletion of 4,551 base pair in the YRF 23, 24, and 25, then deletion of 4686 base pair in third region, that's called YRF 30. Then there is a huge deletion of 5,889 bases. There is a complete deletion of the YRF 116, 117, 118, 119, 120. Then fifth deletion that is occurred of 2,346 base pair. That's called the deletion of 168, 162, 63 base pair, YRF. Then we compared our old strain genome sequencing with all the old genome sequences of WS available in the world in the GenBank. So that is the Indian old strain. We have the 2,92,967. This is our new strain, the 2,79,797 base pair. So this is the Thailand strain. That is the Taiwan strain. That's called the largest genome. That is China and South Korea, Mexico, and Australia. So we can see, actually we have seen that is a 5 region deletion here. So there is no deletion here. We also compared the deletion here also here compared with other things. So not only with our sequencing, we also confirmed with PCR also. So actually totally 10 wire has been deleted. It's a major deletion genome of the new strain of the WSSV. So for each YRF we have designed the primer. So we have confirmed individually for each YRF in the oldest new strain of the WSSV. So here we have used that VP28. VP28 is a marker for both new strain and old strain. You can see the amplification of VP28 in both strain. Here you can see this is old strain we have amplified. That is YRF 24. There is no amplification. It means there is a deletion. So like that we compare. We have done the piece analysis for all the 10 YRF. You can see all the 10 YRF has been deleted here. As you can see the fourth land, there is no amplification at all. That means there is totally that 10 wires have been deleted in the new strain of the WSSV. Then another important task in our diagnosis is then we have to how to detect this old strain, new strain. So then we have to differentiate the old strain and new strain in the samples. Suppose we don't know whether this animal is infected with new strain or old strain. So we have designed a primer based upon the 10th position here, this primer. So this primer, the animal temperature is 58. So this primer can amplify the old strain. So the 2748 amplification. So in the new strain, it will amplify only 4402. Suppose that this primer set amplify the infected animal, have produced a product force there, that means that the animal is infected with the new strain of the WSSV. Suppose if the primer set amplified the PCR product of the 2748 base pair, that means that this animal is infected with the old strain. So now we can easily differentiate the old strain, new strain of the WSV by using this methodology of PCR. Oh, one thing I want to show the slide. Okay. So next we'll come to our control and preventive measures. So any practical method to eradicate or inactivate the WSV in the culture system would be enormous. So here I want to tell one more thing here. Actually, so this new strain is only unique to India, nowhere in the other countries. You know, another important thing is it is highly virulent when compared to the old strain. So why it is more virulent? Because that genome size is very less when compared to, actually this is the shortest genome when compared to the other genome of the other strains of the WSC available in the world. So because of the shortest thing, the replication time is very less. Definitely old strain also pathogen and causing mortality 100%. But here that 100% mortality time duration is very short. Suppose it is 48 hours means it is 40 hours. So both strains are pathogenic, but it is when compared to old strain, it is a little bit more pathogenic. Now another important thing is now that is old strain totally replaced by the new strain. Now there is no old strain at all in the Indian system now. So so we don't know. Actually, that is after introduction of the Vaname, this virus is mutated and started causing the mortality in the Vaname. See, I told earlier there was no mortality at all between 2010 to 2015. I cannot say no mortality, but not a significant mortality. So after 2015, that means after occurrence of this new strain, there was a regular mortality due to WSV in the Vaname. So yeah, there is no, suppose if any practical methodology is available to control this virus will be very useful for the farmers, not only the farmer, even industry. So no drugs to treat WSSV infected shrimp. That you may be knowing that from we are suffering from that now coronavirus infection. So vaccination is only possible for coronavirus, but here vaccination is not possible because shrimp has very simple defense mechanism. That's called innate immunity. So actually in our laboratory, we have tried different strategies. Actually, that is our work only, research work. We have done a lot of research work to develop some good strategy to control or prevent the WSV infection in the shrimp. So actually we screened various antiviral plants. So actually these are the so many antiviral plants we tried. Just like our the Siddha people, Ayurvedic people are trying with products. So it's also very useful to control, to treat the corona patient. So here also we tried that antiviral plants. So we selected that antiviral plants, anyway, I'll come to later. Then next strategy, we used the use of recombinant proteins of WSV as immunostimulant to enhance the immunity of the shrimp against WSV. Then application of RNA technology to prevent the WSV infection in shrimp by silencing the expression of the WSV, VP28 gene of WSV. So these are the three strategies we developed in our laboratory. Some of the things has been commercialized, some of the we completed the field trial. So some of the things to be handed over to the industry for field applications and commercialization. So what is antiviral? So you should know now because in the scenario of the coronavirus now. So any antiviral, any compounds having some to control or inhibit the viral virus is called, we can call it as antiviral compounds. So there are three types of antiviral compounds. One is virucidal compound. That means this is a compound directly inactivate the viruses. So this is called as virucidal agents. So what is the antiviral compound? The antiviral compound inhibits the viral replication. We can say it's the antiviral compound. The immunomodulators. Suppose some of the immunomodulators, some of the compounds which enhance the immunity or induce the immunity of the host, and also that the immunity control the viral viruses and also eradicate the virus in the body. So that is the reason. So the people are asking us to take the more immunostimulants like turmeric and other pepper. These are immunomodulators. So to enhance the innate immunity of the animal as well as innate immunity of the human being also. So what is the methods of this compound attacks the virus? This time is consuming. Okay, I'll go fast. So the prevention of the viral entry. Okay, here is skip all the things. So that actually that is we tried with so many viral plants. So these are the viral plants we selected based upon the antiviral activity against the other human viruses or animal viruses. So these are the viruses we have tried. So based upon the virus, based upon this our work, we one of the plants showed a very strong antiviral activity against this weight percent of virus. So then we laboratory. So we have done so many tests. We have done in the laboratory. So we have tried this viral compound, antiviral compound prepared from the plant. So by IM route as well as oral route. So we got very good result. So this work is funded by the DBT. So we started the work in 2003. So we have conducted many trials, laboratory trials as well field trials from 2003 to 2015 by both the IM route as well as oral route. So we have also confirmed the inactivation by PCR and RT-PCR and Western blot. So we found that there is no viral particles after treatment of the infected animal with this our plant compound. Confirmed with histopathology. So actually this our laboratory as well as field experiments revealed that so this plant compound has the dual effect. The one is the virucidal effect. Actually this compound directly inactivated the WSSV. We have confirmed with in vitro assay using the crab heart explants. So it's also acts as a immunostimulator. Actually this we have analyzed the various immunological analysis. So we found that there is a high significant increase in the level of the prophenol oxidase. So this prophenol oxidase having strong antiviral activity with so many viruses. We also that we observed a significant high level of the prophenol oxidase after treatment with our plant compound. So this high level of the prophenol oxidase may help the animal to protect the shrimp from the WC infection. So our observation also supported by the previous work also. So Selfie and Buffon 2006 they found that so prophenol oxidase inhibit that virus called as in so many several vertebrate viruses in vitro as well as in vivo. Even the high level of the prophenol oxidase protects the HZIA from fatal infection caused by the nuclear polyhedroviruses pathogenesis. So these are the reports of the previous workers which supports our observation of that efficiency of the prophenol oxidase. It controls the WSSV in the infected animal. So actually we also produce the large scale of this compound in our laboratory. Then we large scale production helped us to conduct the many field trials with help of the company. So we have transferred this technology to the company. Then the company has commercialized the product in the form of Imuzone. So still the product is in the market. So till today, that company has sold more than 45,000 liters of this compound worth of more than 4.5 crores. So this compound is having full demand among the farmers. So continuously the farmers are using to prevent the WSC infection in the farm. So every year they will give the sale value to our laboratory, how much they sold for this Imuzone compound. So in addition to that, we got a project, the DBT project very recently. So we have just 2-3 years back. So we ourselves we have conducted the meeting, we have conducted the field trial with our plant compound. This called a farmer. It's called as this farmer is from the Mungamur, Nellore district. So regularly we hand over the plant compound to the farm. So they will apply in the feed, then feed the animal. So then we observe regularly. So this is our advisor that we hand over the one time. So regularly visit to the farm. So one time that our advisor also came to the farm. So he hand over the plant compound to the thing. Actually, that is this is the harvest. We have taken that one. Actually, we conducted 3 trials. So that all three trials we got very good production. Actually, this is the first production that's called 2016 to 17. That is called a 3 months program without any of our intervention, without a herbal compound. So we observed his activities. So we stocked 1.2 million PL. Then there was a WSS infection as well as EHP infection, and we observed. Then we harvested because of the WSS infection. Between that body weight is 8 to 15 gram. So the survival also very low, 33 to 60%. So the production was only 11,500. So his production, he has been doing that farming for the past 15 years. So he never crossed the production not more than 11,000. So then we entered. Then we started giving that our herbal immunostimulant to the farm. So this time we stocked 1.2 million. So then we regularly that animals fed with immunostimulant feed. That herbal immunostimulant. Then we stocked DOC, day of culture is 116 days. That body weight is increased 15 to 33 grams is body weight harvesting. So we got 18,000 kilogram after 3 months. So here next crop also we helped by providing the herbal compound. Then third crop also here. So just you observe here, so the production just become doubled after our intervention by giving our plant compound. There is no infection at all. So only we observed only the one part and one time that also cleared. So that this is also one of the very important proof. We also submitted the report to the DBT. So that was a very excellent result we got with this farmer. So not only that, so we also took the animal from the farm, treated and untreated farm. So we brought to our laboratory. Then we also challenged with WSSV. Then we got a very good survival. That's called after challenging, we got 86% survival. So 66 to 86% survival we got in all the three crops. So we got 100% mortality in the untreated part. Again, we have come back to the same slide of the previous one. So how to manage and get the successful production? So just like this thing, same three factors: pathogen, host, and environment. So avoid the pathogen entry to the system. You'll get the good success. So how to avoid? So understanding the pathogen first. So we have to understand the pathogen. We use the most sensitive diagnostic tool for screening the brooder and seed, as well as live feed, and also monitor. So water treatment, reduce the exchange, eradicate the carriers and reservoir and other hosts, proper disposal of the dead streams, and disinfect and contaminate water. So all these activities will help to prevent the entry of the pathogen into the system, and you will get the successful production. Regarding the host, stocking pathogen-free seeds, that is very, very important. Avoid overstocking to reduce the stress, feed, nutritional feed, avoid overfeeding, use viral and as well as herbal immunostimulant, use antiviral compounds, regular sampling to monitor stream, and minimize. Then keep the environment also good by cleaning the water, cleaning water, everything. So in that case, the disease can be managed. So I am very much thankful to the committee members as well as the Department of Biotechnology. Most of our work has been funded by the DBT. So ICR, NFD for management of Hakim College, my teachers, including Altaf sir, he is our role model for us, then by all my research scholars. Thank you.
Thank you, sir, for the wonderful lecture.
Thank you, Dr. Shaul. It is a wonderful lecture. Really, we enjoyed it. Only one clarification I would like to have from you, Dr. Shaul.
Please,
can't we find some such compounds for the corona?
No. So it's there. Some of the plant compound
maybe it is there.
Actually, we already experienced the antiviral activity of the plant compound because we have very good resource. So already that so many literatures are available. Not only that scientific literature, even Ayurvedic also. We have so many Ayurvedic books here. So we have things. But thing is, the work should be. We have to carry out the research work on all the compound. So actually, what we have screened 20 plant is based upon the antiviral activity of the other viruses. So among the 20 plant, one plant worked very well against WSSV. So that we used. So like that, coronavirus also we can have some plant compound. There's no doubt at all.
In this regard, I feel that there may be many bioactive compounds in the marine resources that can also be probed. So thank you so much.
Yeah. Any questions? I'm very happy to answer.
Sir, thank you very much, sir. Myself, Principal Bhupati Bhaskaran here. Your, a lot of research what you is going on, and the test methods what you have adopted and getting things done is very much interesting to hear. And more and more, I think the awareness has to be brought up because many are still not aware, especially in Tiruvallur district and in Kanchipuram district also. Vellore, of course, maybe they are aware a little, but these two districts, even Chengalpet, I could see some, but they are not making use of the labs properly to get their problems solved. So somewhere the message has to be passed, information has to be passed through WhatsApp group or some method, the farmers can be aware of, sir, and contact you in time and get the remedial actions as that of in Andhra what you faced, and you have given a very good solution for it, sir.
Yeah, no, actually, that is what you told, that Tiruvallur, Viluppuram and Kajum districts are in our surveillance program only. So regularly we visit all the farms, and also we help the people also. But who are all people interested, we help, because those people are not interested, then what we can do?
Yes,
even
I
for
do
example,
understand.
even for example, even that the current scenario of the corona, so even the most of the vaccines are very efficient, but the people are still hesitating to take the vaccine. So even though
Correct,
that is
sir.
the government is making so many awareness program, in spite of that, the people are not coming forward to take the vaccine. So same problem.
Yes.
Even here you can see the farmers, same problem everywhere. That there is because of the what we can say. So maybe illiteracy, or we don't know actually.
Correct, sir. Anyway, I do accept your point, sir, and it has been a very valuable session, I should say. Thank you very much, sir. Thank you, sir.
Any other questions from the participants?
It's very informative. We are so fortunate to have you, lecture, sir. Thank you.
Okay, thank you for all.
Thank you, Dr. Shaul. It's a really wonderful lecture. We enjoyed it, and it's very informative. And people who saw it, even those who are not involved in the aquaculture, they might have learned a lot about the virus and its details. Thank you so much for spending your valuable time.
Thank you.
Thank you, sir. One announcement. The participants who have registered for oral presentation are requested to be ready with their slides. After the third session, we'll start the oral presentation. Moving forward to the next session, the next speaker is Dr. P. Sandanam, Associate Professor from Department of Marine Science, Bharathidasan University. He is having 20 years of teaching and research experience, which specialized in marine plantology and aquaculture. He has published 230 research articles in reputed journal and 81 books with cumulative impact factor 78.369, and the total number of citation is 1677. In addition, he has submitted 46 sequences in gene bank. He has completed 8 major research projects in different major government funding agencies. During his research period, he has developed technology for the culture of marine copepods for the first time in India, and also he has developed immobilized microalgae photobioreactor for the first time in India. He has established microalgae culture facility in Bharathidasan University. He has actively involved in extension or outreach activities like providing training and awareness on hygienic handling of marine fishes, fish value added product development, culture of live feeds, culture of marine food and ornamental fishes. With this short introduction, I would like to invite Dr. P. Sandanam sir to deliver the lecture. I welcome you, sir. Over to you.
Thank you, madam, for nice introduction. I request Professor Sahul sir to just log out his presentation, sharing his presentation, so that I can able to share my PPT.
Sahul sir, please stop the screen sharing, sir.
Yes.
Thank you, sir. Thank you.
I hope my PPT is visible.
Yes, sir. It's very clear, sir.
Yeah. May I audible?
Yes, sir.
Okay. So, very good morning, one and all. It gives me an immense pleasure and privilege to be a resource person in this very important webinar. The topic is mariculture. First of all, I would like to convey my sincere regards, respects, and thanks to the eminent professor of planktonologist, Professor Dr. K. Altaf sir, who is an instrumental to given me this opportunity to share my experience in the field of copypot culture and his role in aquaculture or mariculture. So I would like to extend my sincere thanks to the authorities of this AMET University, especially the principal, registrar, vice chancellor, and the head of the department of the Marine Biotechnology department, and all the faculty members. So let me start my lecture. My topic of today's lecture is the coffee flat, a novel approach towards the sustainable mariculture. So the coffee flag is nothing but copypot is a major metazoan inhabiting in the marine ecosystem, not only marine, in all aquatic ecosystem. So this is actually one of the historical picture. As whenever I used to give the lecture like this, I keep this is a first slide. Actually, this photograph was taken by a famous South African photojournalist, Mr. Kevin Catcher, when he was visited Sudan. You took this picture, so for which he has received the Best Photography Award. But after recipient of this Best Photography Award, there are a lot of criticism about the Kevin Catcher. Do you know? So after the criticism, he has committed suicide. Do you know why he has committed suicide? Actually, when he was visited Sudan, he took this picture. There is a story behind this picture. There is a Sudanese girl child. Actually, she is trying to move towards the UN camp, United Nations camp, where the meal is served. Unfortunately, before reaching the destination, she is almost dying, and just behind the girl child, there is a vulture waiting to eat after the girl child died. This is the real situation in all undeveloped countries and some developing countries like even in India. Because why I am telling, we are talking India is going to be a superpower or going to be excellency in various aspects like remote sensing, satellite and computer and so on. But you know, there are 15% of the Indian population every night sleeps hungry. There are 194 million Indians are undernourished. There are in the world, out of 117 countries, the India is identified as one of the hunger country which is ranked at 104th place. But these issues, which hardly finds its place in media reports, prime time, or in sensational or breaking news. This is a real situation about India. According to World Health Organization, in India, there are 70% of the children under 6 months to 5 years old are in anemic. And similarly, the children under the age group of 5 years, there are 20% children are under acute undernutrition. And 48% of the people in India, they are under chronic undernutrition. And the 28% of the men and 33% of the women in India, they are under malnutrition. And very seriously, you see there is a report, nearly 56% of the pregnant women, they are in anemic condition. So this is the real situation in India, not in undeveloped countries like any African countries. Even in India, this is the real situation. This report is given by the World Health Organization. Okay, now I have a question to you. Is there any solution to solve the malnutrition, hunger, poverty, and so on? As a marine biologist, as an aquaculture scientist, I can suggest there is a two sector which is going to be a solution to solve this malnutrition, hunger, or poverty. One is agriculture, the another one is aquaculture and fisheries. You may ask how the aquaculture or fisheries is going to be a solution for this eradication of malnutrition, poverty, or hunger. As you know well, the fish is the cheapest protein source available on land. And similarly, the fish is a good source of omega-3 fatty acids, and fish is one of the important entrepreneurs' livelihood option. So by providing the fish, we can eradicate the malnutrition, and by producing more fish in confined environment, in open ocean or in aquaculture pond, we can able to generate lot of incomes and employment. So this will be solve the malnutrition, hunger and poverty issues. However, unfortunately, the recent report says that due to overexploitation, global warming and pollution, the fishery resources from the wild, that is in the ocean, is declining like anything. So according to UNEP report, they have stated that in 2050, if the current situation is continued, current situation means overexploitation, global warming or pollution continue, in 2050, there won't be a single fish in the ocean. If no fish in the ocean, from where we can get the cheapest protein source, that is fish. So the potential alternate source is aquaculture or mariculture. So you know when aquaculture or mariculture is nothing but culture of marine organisms or aquatic organism in confined environment is called aquaculture or mariculture. So ultimately, if the production decline in the wild, the alternate source for the supply of fish and other fish food commodity is aquaculture or mariculture. However, again, unfortunately, the aquaculture or mariculture sector is also facing several threats or issues. The disease is one among. I'm very happy Dr. Sahul sir has given very good lecture, and the first lecture was given by Dr. Kailasam sir, who is my well-wisher. Both the lectures have given a basic information to my lecture. So the diseases is a major threat in aquaculture and mariculture aspect. So because of diseases, the farmers, they are losing their precious crops. Okay, to cure the diseases, the farmers, they are using some chemicals and antibiotics. Okay, of course, this using the chemicals and antibiotics, they have given very good, some partial success result. Unfortunately, the chemicals and antibiotics which the farmers are using in the culture system, that will create a side effect to the cultivable organism, and all the residues that absorb the chemicals and antibiotics can be transferred to the consumers through the fish. So that the consumer, the human being, by eating the fish which has the residues of antibiotics or chemicals, so because of the residues, we are getting some health disorder or health risk. To overcome this, the farmers, they are moving towards the organic farming. Organic farming is nothing but using the organic fertilizer or the organic manure, we can able to produce the cultivable organism. This will give some partial amount of success. Again, in organic aquaculture or organic farming also, there is an issue called eutrophication. The eutrophication caused by left out organic waste. The left out organic manure or fertilizer, it will create another unwanted issue called marine pollution or aquatic pollution, or otherwise called eutrophication. So this may not be a sustainable system. In this context, in this line, now there is one farmer in Thailand, not even a scientist or not even a professor, a farmer from Thailand, he has developed the technology called coffee flag. The coffee flag is nothing but is the new aqua farming technique introduced in Thailand, which was developed on the basis of technology bioflag. Advantages and top particles is use natural food alone. There is no any industrial food use. Here, using natural food, along with the natural food, there are some bacteria, probiotic bacteria, which will taking care of water quality in the culture system, and the natural food will be a very good nutritionally superior food. So using the natural food such as coffee pod and using the probiotic bacteria, the farmer is doing the culture, is culturing the shrimps, vanami, using this novel technique called coffee flag technology. So for next 45 minutes, we're going to explain how the coffee flag technology or coffee pod is going to be a superior food in aquaculture or mariculture system. Okay, what is coffee flag? Coffee flag is the most advanced technology to the aqua farming industry that provide natural live diet. There is nothing but coffee pod for larvae prior to the stocking. And what are the benefits out of getting out of using this technology? This coffee flag technology increase the pond water stability as it is natural food, and it enhances the survival of the cultivable organisms. Why? Because I will explain later in coming slide why the coffee pod is enhancing the survival and growth in cultivable organism. And this technology, it enhances the growth of the cultivable organism, and it gives high profitable to the farmer. And this technology, coffee flag technology, totally sustainable. It is totally sustainable, not destructive to the environment, as the coffee pod is natural organisms. Okay, So what is the need for co-flat technology? We have a traditional way of farming using the industrial food. We are growing the shrimp, we are growing the fish in marine system or in any confined aquaculture system. Then what is the need for co-flat technology? Because this co-flat technology, the highly essential to produce more aquaculture products or mariculture products without significantly increasing the usage of the basic natural resources such as water and land. With a minimum land, with a less amount of water, we can produce the cultivable organism by using the coffee pod as a natural feed. And this technology to build up systems providing an equitable cost benefit ratio. This is very important. That the equitable cost benefit ratio to support the economic and social sustainability of the farmer. And the last one is to develop the sustainable aquaculture system that will not damage the environment. So by adopting this technology, the farmers can able to produce more crop with less investment without using chemicals or antibiotics, and even there is no need of any water exchange if you adopt this technology. So this technology is totally sustainable. Okay. So with this background, these are some advantages of coffee flat technology. Here, since we are not using the industrial food, pellet feed, that pellet feed is a major source of pollution. The leftover feed will be a major source of ammonia in the culture system, which will create a water quality problem that will ultimately affect the cultivable organism. Here there is no industrial feed, there is no chemicals, or there is no any antibiotics. It is totally natural way of farming, and it will give very good benefit yield to the farmers. So this technology is so totally sustainable and environment friendly. So now hereafter I'm going to explain how the coffee pod is suitable feed when compared to any other feed available in the natural system. Okay, so this is a food source in coffee plant technology for shrimp and fish. The feed used in this system mainly grain and natural food bioflat. There is nothing but coffee pod. The coffee pods are nutritious and suitable for the growth of fish and shrimp. In addition to that, here we are using microbes, some probiotic bacteria, which will taking care of water quality in the culture system. So by using this probiotic bacteria, copipore and ampipore, we can combinedly, using this natural feed and bacteria, we can grow the cultivable organism without any disease outbreak. That is the potential advantage of this system. Okay. So before going into this co-flat technology, how the coffee plant technology increasing the production, let me explain briefly something about the coffee pod. So this is a picture. The coffee pod actually, it is available everywhere. Wherever water is there, the coffee pod is there. And so far, more than 14,000 species of coffee pods are reported, and there are 10 suborders among the coffee pods. Among the 10 suborders, the 3 suborders such as Clanida, Cyclopoida, and Arbactida being considered as a live feed since it has free living coffee pods. Okay, yeah. See, we have rotifer, we have Artemia, we have other live feeds. How far coffee pod is superior than existing live feeds? So let me explain. The coffee pod is rich in protein, which is a body-building biochemical component, and it is rich in digestive enzyme. If you feed your fish or shrimp with the coffee pods, since the coffee pods are supplying digestive enzyme, the fish, they can easily digest that food material within a short period of time, and they used to feed again and again within a short period of time. So which will increase the growth of your cultivable organism. And coffee pods are rich in essential amino acids, and it's an excellent source of highly unsaturated fatty acid and arachidonic acid. See, the people, those who are working in aquaculture, especially nutrition field, they are always looking for feed which supply more protein and omega-3 fatty acid or highly unsaturated fatty acid. So, fortunately, our coffee pod is having excellent profile of nutrition, very particularly the highly unsaturated fatty acid is very rich in coffee. Even compared to enriched Artemia, the naturally growing coffee pod is having sufficient highly unsaturated fatty acid and arachidonic acid, which is being considered as a body-building fatty acid. In addition to that, the coffee pods are having antioxidant activity. It is rich in astaxanthin, vitamin C and E. In addition to that, the coffee pods are rich in minerals. So always the fish or shrimp larvae is required, it is available in the coffee pod. So there is no doubt that in nutrition point of view, coffee pod is very superior. Okay, see, I'm not telling blindly, We have analyzed all the biochemical components of our copepod, that protein, carbohydrate, lipid, and all components are, always higher than the artemia and rotifer. You can see here the biochemical profile, and we have analyzed the highly unsaturated fatty acid content of copepod. Then compared to artemia and rotifer, the copepods are showing very good HUPA content, which is highly essential for the growth and survival of the cultivatable organism. And the essential amino acids also very rich in copepods when compared to artemia and rotifer. Okay, actually I asked one of my PhD scholar, Mr. Raju, to analyze, to just compare the HUPA content, highly unsaturated fatty acid profile of the unenriched artemia anomaly, enriched artemia anomaly, and unenriched copepod. Surprisingly, the unenriched copepod, that is naturally growing copepod, is showing very good fatty acid when compared to even enriched artemia anomaly. So the farmers, those who are doing farming, their shrimp production or fish production, they are always doing in hatchery, before feeding their larvae, they are enriching the artemia anomaly or rotifer with commercially available emulsifier, which will increase the cost of production. So if you use the copepod to feed your animal, there is no need of any additional enrichment, which will increase the cost of production. So it is clearly shows that the copepod is having very good nutritional profile. And we have also analyzed the carotenoid and astaxanthin content of our copepod. When we are analyzing this content, the pigments serve as a very good antioxidant precursor. If you feed your larvae or adult fish or shrimp with feed which supply more pigments like carotenoid and astaxanthin, the immune system of the cultivatable organism is enhanced. Once the immune system enhances, it will escape from the most of the diseases, especially specific pathogen-free diseases may not be occurred in the system if you use the copepod as a natural feed in your aquaculture or mariculture system. And we have analyzed the astaxanthin content of artemia anomaly and copepod. So the copepod is always showing high astaxanthin content than the artemia anomaly. Okay, and actually there is one problem with the copepod. The copepod is one of the vector, is a carrier, virus carrier. It is reported in some places, but fortunately in India, so far we have not come across this issue, so that to justify the farmer or to justify the funding agency, actually without justification, I can't claim my copepod is super feed. Okay, so that we have analyzed all the parameters. We have analyzed the antimicrobial activity of copepod and copepod fed shrimp larvae, and rotifer, artemia, rotifer fed, artemia fed shrimp larvae. So surprisingly, we have got very good result, both copepod and copepod fed larvae showing very good antimicrobial activity against the listed pathogens here. So it shows clearly that if you use copepod, it enhances the antimicrobial activity of your animal because the copepod supplying nutrition profile so that the immune system will be developed. Okay, and another advantage with copepod swimming pattern is triggered the fish larvae or shrimp larvae to feed over the copepods. Okay, then see, this is very potential advantage. If you talk about the mariculture, especially most of the marine fish, just hatched marine fish larvae, the mouth size is always smaller than the live feed what we are using nowadays, that is rotifer and artemia. So the rotifer and artemia anomaly is size-wise always larger than or bigger than the mouth size of the marine fish larvae. So here the size-related issue is coming into the practice. Because of the inappropriate size of the artemia and rotifer, the marine fish larvae, they may not be able to feed over the rotifer and artemia, so that the survival is very low. However, if you use copepod in your system, since copepods are providing a wide spectrum of size, it has 12 stages, six nauplii stages, 6 copepod stages, and adult. Each and every stage, it has their own size, so that if you use the copepod nauplii, especially copepod nauplii 1, 2, 3, it is ideal size to feed your marine fish larvae. So the size related issues will be solved if you use copepod in your culture system. Okay. Then another important advantage, the movement pattern. The copepod, it has different movement pattern. It has zigzag movement, sharp gliding movement, and jerking movements. You know, the marine fish larvae, evolutionarily, they adapted to actually prepare the feed which is very active. And the marine fish larvae is evolutionarily adapted to just their feeding behavior. They can chase and feed, take their feed. This is the adaptation the marine larvae is having. This the movement pattern of the copepod is stimulate the larvae to feeding over the copepod. This is another advantage. Very recently, there is one article published in Aquaculture Journal. Copepods are emitting pheromone. I don't know. The pheromone is nothing but is a type of hormone. Is a smell. Okay. So for pheromone, for smell only they are using the fish oil or fish meat as a source of protein or a source of omega-3 fatty acid or source of smell. The moment if you just give the copepod in our culture system, pond or tank system, since the copepods are emitting pheromone, the larvae, the fish larvae, because the fish larvae, their sensory system may not be developed in the early stage. But through the smell, the fish larvae, they can identify the feed and locate the feed and feeding over the copepod. This is another advantage behind the copepod. The last one is, very recently, it is discovered that copepods, some species of copepods are bioluminescent organisms. What is meant by bioluminescent organism? The organisms emitting light. So during night time, actually they just hatched the fish larvae. They're poor, it has poor vision. The vision is very poor. They may not be locate their feed. However, since copepods are emitting light, during night time, if you feed your fish larvae with the copepod, since copepods are emitting light, the fish larvae, they can easily identify the copepod and meal over the copepod. And these are the potential advantage behind the copepod. Since the copepods are having several advantages as an alternative feed, our group, we have started working on developing the copepod culture. Okay, so we are collecting the copepods from different ecosystems like open sea, estuaries, mangrove water all over India, and have started isolating the species. And after isolation, we have identified some potential species by analyzing their biochemical profile, and we have developed the culture. So actually, this is the way for collection. We are using 150 micrometer. The ideal time for copepod collection is early morning or late evening. Once collection is over, we took the samples to the laboratory, provided with vigorous aeration by using the battery aerators. After reaching the laboratory, we actually fabricated one superimposed sieve. This is nothing but is a grading, because if you use 150 micron mesh for collection of copepod, there will be a fish larvae, there will be a shrimp larvae, there will be a rotifer, there will be artemia. It's okay, but our aim to isolate the copepods. So size-wise, we can grade the copepods using different sieves. So for example, here we are using 500 micron mesh, 190, 140, 90. So based on the size, we can segregate or grade the copepods and take the graded samples into the petri plate, sit under the microscope, identify the species level using the available manual or monograph or taxonomy key. After isolation, after confirmation of the species, we have isolated different species. This is one Nanocalanus minor. It's a common species in India. This is Paracalanus parvus. This is Pseudodiatomus anandali. And this is Nithogonophanes. For the first time, actually in India, we have reported this species from Velar estuary. Actually, this particular species is having very good astaxanthin content, and it is ideal feed for shrimp larvae, adult or shrimp larvae. And then we have Tisbe species with us, Eutropin acutipras, Aithona rigida, Aithona dissimis. Very recently, we have developed the culture for this species. I will show you the video of this particular species. Then Tigropa species. Okay, so after isolation, we have started our culture with a small conical flask or beaker. This is a culture facility, stock culture facility established under the financial support of DBT. I should thank DBT because they are the major funding for our all projects, especially on copepod culture. So, for isolation of copepod, there is no need of any sophisticated instruments. Only with a simple needle, simple brush, simple stempel pipette, you can take the diluted sample in the petri plate, sit under the microscope, you just locate the species individual and keep away the other individual. You will gently pick up one individual, whether it may be male or female, or it may be gravid female, egg-bearing female. Like that, you have to isolate the known number of copepod individual, maybe around 100 numbers, which include male and female. You can isolate. That isolated copepods can be transferred to the initially test tube or conical flask or beakers. Okay. And after isolation, we have to feed the copepod. Actually, for stock culture, maintaining stock culture, there is no need of any auxiliary provision. The organ is the best feed to culture the copepod. Okay. So we are using different microalgae such as Chlorella marina, Donaliella, Isocrasis, and so on. So algae at the standardized concentration, we are feeding our copypods and we are maintaining our copypods without any contamination. Okay, so this is the stock culture facility in our university. And okay, see, you may ask, sir, you are telling your copypod is having this much advantages, nutrition and movement pattern, it is having enzyme, pheromones, and so on. Is there any barrier in copypod? Is there any problem in copypod if you take that copypod to the commercial production, commercial culture? Yes, there is one disadvantage with copypod. Getting high density in copypod, still it is challenging because the farmers, they are producing millions of larvae. To feed the millions of larvae, at least we have to provide billions of copypod as a feed. To that, we need high biomass production technology. For that, recently, I have received one project from DBT. I should thank Dr. Shahul sir, who is the task force member of this committee, for sanctioning this project. Because the DBT, I should thank him. They are highly supporting me for the development of this particular copypod species for in commercial scale. So this project that is on selective breeding and induced breeding. How we are going for a selective breeding for shrimp or fish, similarly induced breeding for shrimp and fish. Same way, by inducing some parameters and by selecting the potential brooders, okay, the potential G0 or G1 generation, instead of going for random selection, we can select actually the high egg producing female and the high sperm producing male. So we will select the potential individuals and stock the potential individuals, selected individuals in the confined environment, and then we provide the suitable condition that the male and female will undergo for mating and they can release the more eggs. That is called selective breeding. So in selective breeding, there are two methods. Two selective breeding is there. One is normal selective breeding, other one is cold selective breeding. So the normal selective breeding is nothing but just maintaining in 28 degrees centigrade temperature, and the cold selective breeding is 18 degrees centigrade. So by just inducing the animal, I mean maintaining the temperature, the cold selective breeding is giving very good result in our experience. We have got very good egg production numbers. Now very recently we have come out with high density production. I will show you the video in this coming slide. So by adopting the selective breeding, we can able to produce the copypods in high density. And suppose if farmers want copypod continuously, for that actually we went for induced breeding. As like how we are adopting the induced breeding for fish and shrimp. Similarly, by inducing the environmental conditions like heat induction, cold shock, salinity shock, pH shock, light shock, photoperiod shock, and oxygen shock, by inducing these water quality parameters, we can stimulate the male and the female copypods to go for mating, and immediately they can undergo for reproduction, and the female within 36 hours they can release the eggs. Similarly, we also experience with hormonal induction. Using the hormone, we can just inoculate the hormone directly into the copypod culture tank. Since the copypods are filter feeders, they can take the hormone, and once it consumes the hormone, it will stimulate it for mating and produce the eggs. Similarly, we can, by using the microbial probiotic, microbial adjuvant, that is called microbial induction. Just we have tried with the Bacillus subtilis. We enrich our copypod with the Bacillus subtilis. Once it is enriched with the probiotic bacteria, the male and female will induce for mating, and the female will release the eggs. Through adopting this both selective breeding, we can able to achieve the high density, and by adopting the induced breeding, we can able to produce the copypods continuously without any problem. So now we have very good technology with us for the continuous production and higher density production. So this is actually RNA system established in our university campus under the financial support of DBT. Okay, these are the different components and filters and different systems used for the mass production of copypods. Due to time constraint, let me skip some slide because of the time constraint. These are some, we are using sand filter, bag filter, biofilter, UV filter. The seawater will be filtered through a series of filters. From this UV filter, that UV filter water will be stored in the culture tank. This is actually culture tank where the copypod pods will be stocked, and we are using microalgae as a feed. That microalgae will be supplied by using the dosing pump. We will set the dose, how much dose concentration we have to provide the copy pod. Because as our SAR said, if you overfeed, there is a chance of contamination in copy pod. So we have optimized all the parameters, the temperature, salinity, pH, light, photoperiod, which feed is suitable to grow the copy pod, and which concentration is ideal. We have optimized all the conditions. Now we are producing the copy pods in high density without any challenge, so that we can able to supply the copy pods to the farmers. Okay, these are the different filter system and the apparatus and the harvester system we are maintaining. This is actually the copy pod density. You can see with this induced breeding and selective breeding, we can able to obtain very good density in copy pod. This is particularly in Nanocolanus minor, we have obtained this much density. And this is Theracolanus parvus, we have obtained this much density, which include Napki, Copypodites and other. And this is Pseudodiatomus anandali, which is a colonized copypod. Here you can see a small video clippings. This is actually the movement pattern under the microscope, and you can see the density. This is the density which actually we have obtained in the Pseudodiatomus anandali. Due to time constraint, let me go fastly, okay, because a lot of slides are. This is actually the high density of Pseudodiatomus anandali in 10 tanks. In 10,000 FRP tank, we are growing the copy pods. We have got very good result in density and biomass is concerned. Actually, with this, we have validated our copy pod suitability as a live feed within marine fish. I will show you the result, okay. And this is the harvested density of the Pseudodiatomus anandali. You can see the density, how much density we have obtained. Okay, and this is Nitogona affinis density. We have obtained up to 1,80,000 numbers per liter, and this is a Nitogona species, which is ideal feed for shrimp. And this is Ithona rigida. To my knowledge, among the copepods so far we have developed the culture, the Ithona rigida is a very promising species in terms of density, because till date, we have achieved more than 2 lakhs numbers per liter in Ithona rigida. It is highly potential species and fast reproducing species within a short period of time. Even in nutrition point of view also, this is ideal feed. You can see this is a video graph, and this is a Ithona rigida, which comes under cyclopide group of copypod. Okay, and this is a harvested copypod. You can see here, this is harvested copypod density. And yeah, this is actually result we have recently obtained. This actually by adopting the selective breeding and induced breeding, we have got very good result on density. You can see here this videographic evidence. See how much density we have obtained. So this is an Ithona similis, and we have also same density we have obtained in Ithona rigida, Pseudodiatomus anandali, and Nitogona affinis. So this much density we have achieved very recently by adopting the selective breeding method and the induced breeding method. So I'm very happy to present. This is the first time I am showing this video. Even I discussed with our Professor Altaf sir. I just informed him, sir, we have achieved high density recently in copypod culture. Because the getting high density is only major challenge in copypod culture. Now we have come across with solving this issue. Now we can able to supply the copypod in high density available with us. Okay, you can see how much density we have obtained. And this is actually recently, there is another one species, Thermocyclops. We have successfully cultured with high density. And recently, actually, to one of my students, Raju, actually I asked him to work on bioflak technology. So we have developed the amphipoid culture. So now along with the copypod, we are also having amphipoid. See, this is Gamera species. So nowadays the amphipoid is also becoming popular live feed in ornamental and other food fish culture. Okay. So this is this species. This is actually the RAS facility established in our university. And okay. So far we have discussed about how much density we have obtained in copypod culture, what are the advantages are there in copypod culture. Actually, confirm my result, to justify the farmers, to convince the farmers, I have to give valid data. Really, whether copypod is increasing the growth, survival, or biochemical profile of cultivable organism, to convince the farmers, we went for a series of experiments with different fish and shrimp species. This is the one experiment. We did a validation of our copepod suitability with the sea bass, Asian sea bass, lattice calcarifer. See, this is the growth profile of sea bass, lattice calcarifer, 21 days experiment. We validated with rotifer, artemia and copepod. So the copepod is giving very good result in terms of length, weight and survival. You can see here the evidence, and we have also analyzed all nutritional parameters of the sea bass fish larvae which fed on different live fish such as rotifer, artemia and copepod. The copepod fed larvae is showing very good protein, amino acid, fatty acid profile. See, this is the photographic evidence. This is the sea bass actually larvae which fed on 21 days cultured larvae fed on rotifer. This is the same DOC larvae fed on artemia, and this is, you can see here the copepod fed larvae. It has well developed high well developed pins. Even the size is also comparatively higher. And similarly, we have also worked with ornamental fish, Monodactylus argentus, and you can see this evidence. The copepod not only enhancing the growth and survival, copepod is also improving the pigments, the coloration in the fish, ornamental fish. You can see this is evidence. This is the fish, Monodactylus argentus, which fed on artemia and opry. This is the fish which fed on copepod. You can see the difference between both the fish. You can see the copepod fed fish having lot of pigmentation on the caudal fin, dorsal fin, even entire body it has lot of pigmentation when compared to artemia fed fish. So when we analyze this, what is this pigment? This pigment is nothing but astaxanthin. So astaxanthin is enhanced in fish or shrimp larvae if you use the copepod as a feed. Okay, and we have also worked with silver pampano, and we have two different combination of feed, copepod alone, 50% copepod, 50% rotifer. We have validated the fish larvae silver pampano. This is actually one of the collaborative work with CMFRI. One of my PhD student, he did this work, and surprisingly, very interestingly, we have got very good result with the copepod. You can see, this is the fish, 14th day cultured larvae, 14th day larvae, which grown using the copepod napoli alone. And this is the fish larvae at juvenile, which fed 50% copepod, 50% rotifer. So the result clearly shows that the copepod is improving the growth and all eye development and tail and our total body length is comparatively higher than the rotifer and even artemia. So we have also worked with the Pinaeus monodon, tiger shrimp. This is a result, how the rotifer, artemia and comparatively copepod is better. Both the length, weight, survival is always comparatively higher in copepod fed larvae than the artemia and rotifer. You can see here the evidence. The first photograph shows the Pinaeus monodon larvae which fed on rotifer. The middle photograph, this is the Pinaeus monodon tiger shrimp which fed on artemia. The bottom photograph, you can see here, this is the larvae which fed on copepod, especially Diaithona rigida. You can see the rostrum development, antenna development, eye stalk development, even lot of pigments, colorations are there in the entire exoskeleton and the carapace. Very interestingly, when we analyze these parameters, all protein, fatty acid, essential amino acids, pigments are always higher than the larvae which fed on copepod compared to the artemia and rotifer fed one. Okay, we have analyzed all the parameters. The biochemical profile shows that copepod is best to feed to produce our larvae. And we have also worked with astaxanthin content, and we have also validated the suitability of copepod with Vanami, Pinaeus vanami. Okay, and in vanami also we have got very good result on growth and survival and nutritional profile. You can see this is the photographic evidence. Actually, this is Pinaeus vanami, which 21 days cultured larvae which fed on rotifer. This is the larvae which fed on artemia. This is 21st day of larvae which fed on copepod. You can see the difference between the copepod, artemia and rotifer fed one. You can see the antenna and the rostrum is very lengthier, and you can see the tension is well developed and the pigments are very rich. You can see in the intestine also this copepod is increasing the astaxanthin pigment. Okay, so this clearly shows that copepod is improving growth, not only growth and survival, it improving the health condition of the fish or shrimp larvae. And we have analyzed the astaxanthin content in different feed fed larvae. The copepod fed larvae shows very good result, amazing result. Okay, actually this is one of the video clippings I want to show you. As I said during my first slide, slide, there is one farmer is growing shrimp by adopting the copepod technology. This is the videographic evidence. You can see how much density is culturing copepod in the pond system. That copepod will be served as a natural diet. This is a lined pond. Actually, this video I have downloaded from his own website. So see the stocking density. By adopting this copeflag technology, we can go with high stocking density. That is 225 numbers per square meter. That much high density stocking we can go. Okay, see, 9 lakhs per 0.1 hectare area pond, 225 larvae per square meter. See, this is the larvae animal which grown on 22nd day, DOC 22. The animal is so active and so healthy. Do you know why the animal is so active, so healthy? Because the copepod is supplying all essential nutrients to the larvae and the animal, so that they are very happily growing in the system without any problem, so that they are very active and so healthy. And they went for weekly sampling. At the end of this video, you can see the summary, production summary. So they are using both the aerators, long arm aerators and bottom diffuser. Bottom diffuser actually to support the copepod growth, and they are also using probiotic bacteria, heterotrophic bacteria, to maintain the water quality, and the copepod will be a feed for the animal. Along with the copepod, they are also using ampipod. But what the farmer is telling, even compared to ampipod, because they are using only less number of ampipods, but the copepod is they are using more than 70% copepod, which will give very good result in terms of production and yield. Okay, you can see the feeding, the average body weight 8.33 gram at 120 cows. See, the animal is how very neat and clean. But whereas in the traditional way of farming, traditional farming system, the animal is very dirty. It looks like because that the animal appearance will adjust the price of the cultivable organism. So if you go for, you can see how the animal is very neat and clean and healthy. So these are possible only with the copeflag technology. So they are also measuring the flag volume, and the 8 hours less than 0.5 ml per liter. See how the animal is very neat and clean. This is DOC 60. The average body weight is 10.52 gram at 95 cows. Okay. Actually, due to time constraint, I am going fastly. Sorry for that. Yeah, they are subjected for the weekly sampling, and the DOC 67. The animal they attained 12.19 gram at 82 counts. At 82 counts. So this is actually the farmer is doing. Okay, in Thailand, one farmer is doing, and even in India, none of the farmers are adopting this technology, copeflag technology. But in recent times, I am receiving call from some farmers from Andhra Pradesh and even Karnataka, and even from some farmers from Tamil Nadu. They approached me for the copepod culture. If we produce the copepod in large scale, in high biomass, definitely the copepod is going to rule the entire not only aquaculture, mariculture system, because their superior nutritional parameters. You can see this is the copeflag RAS system, which will give very good yield. You can the total feed and the FCR, you can see FCR is very less. That is 1.169 is 1. 1.16 is 1. Survival rate is 84. The total production is in the 67th day, you can see this much capacity yield, 9,260 kilogram of biomass they are harvested. I mean at 0.4 hectare pond, they harvested nearly 1 ton product. So it was obtained with copeflag technology. You can see the animal, how they are very active, very healthy. They are so active, so healthy. Why? Because the copepod is supplying the more highly unsaturated fatty acid along with pigment, so that because of high immune system, high immunity, the animal is very active and they can escape from any disease. So far, the farmer is not reporting any disease outbreaks in the system through this technology. Okay, so what I would like to tell at the last, if farmers adopt the copeflag technology to grow their fish or shrimp, the cultivable organism, it will get some advantages, some benefit. What are the benefits the animals are getting? If you feed your fish larvae with the copepod as a feed, natural feed, it increases the breeding and egg quality in fish. It enhances the stress resistance and immunity to the disease. It increases the embryonic development and growth of fish. It increases the gonadal maturation of the fish. Very importantly, the farmer point of view, the farmer viewpoint, it enhanced, it will reduce the FCR. Very good FCR is resulted in copephylact technology. And fertilization hormone secretion can be increased. As you know, when fertilization hormone is responsible for the reproduction of any animal. Since the copepods are providing nutrition, high nutrition, the fertilization hormone secretion is comparatively higher in animal, so that they can breed continuously, they can produce egg in high numbers, so that we can get more benefit in terms of production or yield. And mortality rate during embryonic development can be reduced in cultivable organisms, so that we are getting more survival rate. Grow in elevate, this is very important news. Is it possible to grow the fish without water? Is it possible to grow the fish in high temperature warm water? Is it possible to grow the fish in ammonia, the water which is having high ammonia level? Is it possible to grow fish or swim in water where harmful light is there, or without oxygen or less oxygen? Is it possible to grow the fish or swim? Yes, it is possible. It is possible. We can grow the fish or swim in warm water where the temperature is maximum, and we can grow the fish or swim with highly ammonia level water, and we can grow the fish with limited oxygen. For that, we have to grow our fish or swim with copepod. If the copepods are supplying more nutrition, since copepods are enhancing the immunity of your cultivable organism, if cultivable organisms get high immunity, they can withstand, they can adopt, they can survive in any extreme condition. That is my take-home message. Okay, and in shrimp, in crustacean, if you grow your shrimp or any crustacean with copepod, it modifies the exfoliation frequency in shrimp. It shortens the molting cycle in shrimp. Because as you know, well, molting is an important growth parameter in shrimp. Copepod shortens the molting cycle in shrimp, so that within a short period of time, the shrimp will attain the marketable size. And it hastens the post-larval development. The post-larval development metamorphosis days is shortens. It enhances the spermatogenesis in shrimp, increase the oogenesis in shrimp. Last but not least, it fastens the embryonic growth. So these are the benefits the animals are getting out of eating copepod. Whereas in rotifer and artemia, these all parameters or characteristics not at all possible. Okay, so with this, what I would like to go forward, there is no doubt that copepod is an excellent field which can enhance the productivity in terms of survival and growth. The farmer definitely they can get high benefit out of their culture system. It is totally sustainable and successful way of aquaculture or mariculture. But there are some barriers are there. In future, what are the future prospects? See, actually, to supply the billions of copepod, to feed our millions of larvae, we have to provide the copepods in high biomass level. For that, we have to develop the commercial production system for the copepods in our country. So far, none of the commercial production system is available. So we are the only group working on copepods. At present, we have nearly 12 species of copepods culture, and we have come out with very good technology for high density production. So the commercial copepod production facility should be established in our country to supply the copepod as a feed or as brooder to produce our fish or shrimp larvae, so that we can achieve our goal. Then the copepods should be bring into value addition. It is possible. There are some developed countries, they are using the value addition of copepods. You can see here I have given some picture. These all live copepod products are available in the market in developed countries, not in India. So like that, we should also have the products like this. So I am sure that in this August gathering, I would like to convey my ambition. Definitely very soon, I am going to bring the copepod products we can able to launch the copyboard product in the market with the financial support of DBT, because the DBT is supporting lot for the production of copyboard. So like this, we should produce the copyboard value addition, which will be a very remarkable milestone in mariculture and aquaculture. And for that, we have to establish the broodstock facility for copyboards. Of course, we have established in our university. So like that, we have to enhance more number of broodstock facility in our country so that we can able to supply the brooder for the farmers, copyboard brooders. And the manpower development of copyboard live feed entrepreneurs. Even in countries like Singapore, Taiwan, Thailand, they have live feed shops. As like ornamental fish shop and edible fish shop, they have separate section for the live feed shop. Like that, even in our country also, we have to establish the live feed shop. So my ambition, before my retirement, I would like to bring the copyboard product, both live product and copyboard cyst, and I want to establish the copyboard shop or live feed shop in our university premises. So these are the things are considered being a way forward. And the establishment of copyboard's live feed shop is a time need of the hour. I am sure that with the financial support of funding agency like DBT, we can come out with the establishing the live fish shops, which is going to be one of the heart of the aquaculture or mariculture practices. Okay, with this, I would like to conclude that there is no doubt that copyboard is a promising field. With the copyboard, we can able to grow fish or shrimp, any cultivable organisms without any disease outbreaks with high profitability. So with this, let me conclude my talk. And these are some news clippings published in the dailies about our achievement. Actually, we have achieved the copyboard technology for 2 species that it was published. We have provided the copyboard strain to some national institutes like SIBA, CMFRI, RTCA, and Frederick Farmer ornamental fish breeder. He came forward and they collected the copyboard from us, and they have given very good feedback. The copyboard is increasing pigmentation and growth. So this is that copyboard distribution launching program. Our former Vice Chancellor launched the copyboard distribution, and at last, I would like to convey my sincere and wholehearted thanks to the authorities of the AMET University, especially the Chancellor, Pro-Chancellor, Vice Chancellor, Pro-Vice Chancellor, Registrar, Dr. Jayaprakash sir, and my teacher and my motivator, my inspirator, Dr. Altaf sir. Actually, he is also working on copyboard, but he is always actually encouraging me, and he is always guiding me in right way to do these things. He has given very good valid suggestion to me how to improve the copyboard culture, how to bring the copyboard product in our country, which will be a very good, remarkable achievement, which will create more production. So thank you, sir, for your advice, continuous advice and blessings. And even I should thank you for the invited me to deliver this lecture. And I should thank our inspiration, Dr. A.R. Tirunavagur sir, who is a former scientist and head of Fish Culture Division, SIBA, and Dr. M. Kailasan sir, because these are the people behind me. Whenever I need larvae for my experiment, the SIBA's larvae, they are readily helping me, and he has helped in such a way so that we have come out with very good findings in the field of live feed culture. And I should thank Mr. Jairaj, who is the senior technician in one of the private hatchery, Marakanam, Marine Hatchery. Whenever I approach him for the seed for our experiment, he is without charging any single paise, he will give. So I should thank him, and I should thank my guru and mentor, Professor Perumal, who introduced me into this field, copyboard culture. And last but not least, I should thank my students, because my students are my support, my strength. So because of our students only, we have come out with very good breakthrough in copyboard culture. I should thank some students, especially Dr. Anand and Mr. Raju. Those are really dedicatingly working even during this pandemic time. Our students are working and taking care of our copyboard and algal culture. So I should thank our students. Last but not least, I should thank my university for granting me permission to deliver this talk. With this, let me thank once again each and every one of you, and particularly I thank participants for your kind listening. I am very happy to answer your any queries if you have any queries. Thank you very much once again.
Thank you, Dr. Santanam. It is marvelous lecture on copyboards, a subject which most of us are ignoring, but
yes,
it's a very vital aspect, and God should help you to achieve more and more in this. And I wish that your product should come out so that we can have indigenous feed for our shrimps and fishes. That will be a big breakthrough.
Yes,
All
sir.
the best, Dr. Sandhanam.
Thank you, sir.
Thank you so much. Thank you
Thank
so
you, sir.
much for your wonderful lecture.
Thank you, sir. Thank you for your guidance and the blessings, because I need people like you. Because I have very good my well-wisher Dr. Arasu sir, my guru Professor Perma sir, Dr. Kailasam sir, and Altaf sir, because they are always encouraging me to do something to this society. Whenever I talk with Professor Altaf sir, he is always talking about sir, co-people should be bring it as a product. That will be a remarkable achievement to the sustainable aquaculture or mariculture. Thank you, sir, for your continuous support and blessings.
Thank you. Thank you.
And in this August actually gathering, I should thank DBT, one of the important, the funding agency, actually going to sanction nearly 4 projects. Around 3 crores they can sanction the project. And very recently, one more project also is going to be released in collaboration with Dr. Sahul sir and Dr. Bright Singh sir. So I should thank all the task force members of the DBT, Dr. Sahul sir and other members for the great support and encouragement. Thank you, sir. I'm very happy to answer your queries.
Sir, myself, Principal Bhupati Bhaskaran, I first and the foremost thank you so much. You know, we all, of course, the nutrition value of the item what you have come up with, and I hope in India this product everyone will start using it, and definitely you have given a big breakthrough in the minds of fish farming and the prawn farming members, and very easily nutrition value for the seedlings as well as the grown fishes and the shrimps that you're going to get means it's something wonderful, sir. Excellent presentation, sir. Thank you very much, sir.
Thank you,
look
sir.
forward a lot from you, sir.
Thank you, sir. Thank you for your compliments.
Though I am a marine engineer and heading the marine engineering team, I've got nothing to do with this, biotechnology. But at the same time, being a farmer and being a background of, animals lover, I have a lot of passion towards it. I always ask Dr. Jay Prakash Well sir, our registrar, whenever he was a registrar to, HOD of biotechnology and all, I used to speak to him of azospilum and small, so many other things. But now, lot more platforms available and more of entrepreneurship can be developed from this, sir. It's something very marvelous, sir. Thank you, sir.
Yes, sir. I'm sure that very soon, with the financial support of funding agency, especially DBT, we can launch the products very soon.
Sure, sir. Thank you, sir.
Thank you, sir.
My best wishes, sir.
Thank you, sir. Thank you, sir.
Thank you, sir, for the excellent lecture.
Thank you, madam. If any questions, you just ask the participants to post their questions in my email so that I can answer to them.
Sure, sir. For further queries, the participants can reach out through the mail, and the speaker ID will be shared shortly. Thank you, sir.
Thank you, madam. Thank you, sir, Professor Altaf sir, and the entire department, Dr. Seminathan sir, and my
Thank you so much, Dr. Sandhanam.
Jayaprakash Well, and controller, my friend Dr. Muttailan. I thank one and all.
Thank you, sir. Thank you for being with us, sir.
So shall I move forward?
Yes.
Yeah. Please.
Moving forward, now we are going to start the oral presentation, and the participants are requested to be ready with their presentations. The participants will be given 7 minutes, 5 minutes for presentation and for discussion. The first presenter is Mr. S. Surya Kodi from C. Abdul Hakim College. Mr. Surya Kodi, you can start presenting your...
Hear you, but I can't able to share my screen.
Now you can able to share the screen.
Yeah. Can all of you able to see my screen? Yeah, okay. Yeah, okay. Good afternoon to all. And I'm going to present the application of rainbow trout cell lines in virological studies and fish viral surveillance. The first and foremost objective of the present study is to test the susceptibility of the rainbow trout cell lines against fish noda virus, Tilapia-like virus, and Cyclodeoxys virus 2. Then, this is the first ever cold water fish cell line developed in India and first reported rainbow trout cell line established in India. So, we make use of this cell line to monitor the surveillance of fish viral disease of cold water fishes using rainbow trout cell lines. Then, the most important objective of the present study is to isolate viral pathogens from disease-suspected fishes. Due to the continuous mortality in the northern part of India, active surveillance have been taken place from January 2019 to February 2020. Then we collect more than 30 samples from the Assam, 7 districts of Assam. Then methodology. Cells were exposed to virus and observed regularly for the any morphological changes. Then confirm the virus in the infected cell lines using virus-specific primers as follows. First, from the infected cell lines, we just isolate the nucleic acid, DNA and RNA extraction. Then from RNA, we converted cDNA conversion. Then we confirm by reverse transcriptase PCR and PCR. Then the cells infected with FNV was collected at different days of post-infection and confirmed as follows. Then the cells collected at different days of post-infection was used to detect Western blot and ELISA using antibody raised against the recombinant fish noda virus capsid protein 42. Then the viral titer was quantified by PCID 50 median tissue culture infectivity dose. The samples collected from different districts of Assam were processed by homogenating with the L15 media without photodomain serum albumin. Then the samples were exposed to the rainbow trout cells and observed regularly as mentioned above for morphological changes. Once any morphological changes were occurred in the cells, then the cells have been taken to the DNA and RNA extraction from RNA as follows. Then the cells infected with the disease-suspected supernatants was collected and analyzed as mentioned above with the respective virus-specific primers designed from our laboratory. The primers and the sequence used in the present study are given in the table as follows. Results. These are the list. These are the 3 cell lines of the rainbow trouts developed in our laboratory. The first 3 cell lines are the control cell lines. The second 3 cell lines are the FNV exposed cell lines. And the third 3 cell lines are the TLV exposed cell lines. The result shows that the RT-PCR results of the fish noda beta noda virus infected cell line. It is clearly indicated that all the 3 cell lines are susceptible to TALV and fish noda virus. This result shows that the Western blot analysis of the fish noda virus infected cell lines. The samples have been collected from 3rd day, 5th day, 7th day, and 10th day. The first image shows that the ELISA results of the fish noda virus infected samples. Group South Indian group array SIC cell line used as a control. And the second image shows that the titer value of the noda virus and the same Sahul Indian group array cell line was used as a control. These are the list of primers used in the present study to confirm the viral infections. This table shows that the fish species collected from different locations of the different districts of Assam with clinical signs including hemorrhages, ulcer on the body, and lesion ulcer over the body. These are the list of 10 viral pathogens screened for the infection. And above in the study, we have detected no virus. These are the PCR results for the disease-suspected samples collected from different districts of the Assam, India. Conclusion. These 3 cell lines were found to be highly susceptible to TLV and FNV, and it has a high ability to support FNV replication. These 3 cell lines should be very useful for propagation of FNV and TLV, and also for the large-scale production of viral diagnostics and vaccines for the benefits of the aquaculture industry. These 3 cell lines would be a useful diagnostic and vaccine production tool in the cold water fishery sector of India. Furthermore, these cell lines would be also useful for surveillance of viral pathogens in cold water fisheries. Thank you.
Thank you. Any questions? If there is no question, we'll go to the next presenter. Or the participant can post questions in the comment box. So now I invite Dr. V. Janaki Raman from Department of Zoology, the National College, Trichy.
Good afternoon to one and all present here. Am I audible to you?
Yeah.
Yes.
Please continue.
Thank you very much. I have the pleasure of presenting my paper entitled Comparative Study of Zooplankton in Different Livelihood Habitats of Tamil Nadu, India.
Sir, excuse me.
Yes, sir.
Please put it in the slideshow mode, sir. We couldn't able to see that full screen mode. It is showing the mobile screen. Yeah.
Sir, I'm using mobile.
Yes, sir.
Shall I start, sir?
Oh,
As
yes.
it is.
Yes, please.
Thank you, sir. As most of the chief guests are introduced about the zooplanktons, let us introduce brief introduction of it. As you know, zooplanktons are heterogeneous, a group of microscopic, free-floating or drifting organisms are found in most of the limnatic as well as the marine habitat. They are served as a secondary producer or primary consumer in aquatic food chain, though they are few micron to several micrometer. Their contribution in aquatic ecosystem are very indispensable role. They are ecologically as well as economically important organism in several ways. Let us see some of them. Zooplanktons shows quick responses to the environmental variation, particularly the physicochemical parameters, and they are used as a bioindicator of aquatic pollution. If there is any variation in the aquatic medium, they shows their quick responses in the form of high density or least diversity. In that way, they are served as a bioindicator of aquatic pollution. And more than that, they are also used as a live feed in aqua hatcheries, most of the finfishes as well as the shellfishes. They are also used as a biocontrol of mosquito larvae. Particularly the copepod-like organisms, they are controlling the mosquito larvae in aquatic medium. And more than that, the planktons are important sources of industrial products such as chitin and its derivatives. In research aspect, the zooplanktons are ideal organisms for toxicological studies, as well as they are also used as a evolutionary relationship organism, because the most of the zooplanktons comes under arthropod family, particularly crustacean. So that so many advantages are there in making a phylogenetic tree of plankton. So based on this, I have made some objective of the studies. First objective is to carry out this density, diversity, and distribution of zooplankton in different limnatic habitat, that is the freshwater habitat of the some certain part of Tamil Nadu, particularly a pond like Pudinagar Pond, Pudinagar Lake, Gundur Lake, Korea River. To study the water quality of Pudinagar Pond, Pudinagar Lake, which is located in the Viluppuram district, and Gundur Lake and Korea River of the Tiruchirappalli, to compile the data of zooplankton for analysis statistically to infer the ecological status of this habitat, to highlight the zooplankton species which are assessing the ecological indicator of aquatic pollution, and also to study the current ecological status of Pudinagar Pond, Pudinagar Lake, Gundur Lake, and Korea River, whether it is polluted or not, based on the presence or absence of the zooplankton. For methodology, water samples are collected for temperature measures. Temperatures was measured by thermometer. Dissolutions were done by Winkler method. pH was measured by pH meter, and other water quality like ammonia, phosphate, nitrate, are measured by using the test kit aquarium pharmaceutical Canada. For quantitative analysis of zooplanktons, 100 liters of the sample were filtered. For qualitative analysis, planktons are collected using the zooplankton net, which are fixed with 5% formalin, and zooplanktons were identified standard methods of Naval and Naval, Rajendran 1973, Dasat and Defe 1995, Ganapati 2000, and Altaf 2004. Quantitative analysis was done by the method of Santanam et al. 1989. The plate 1 shows the different stations of the collected point: Pudanagar Pond, Pudanagar Lake, Gundur Lake, and Koraya River. The result shows the first table 1 shows physicochemical parameters of the different habitat: Pudanagar Pond, Pudanagar Lake, Gundur Lake, and Koraya River. Atmospheric temperatures was minimum in Pudanagar Lake and maximum in Koraya River. Surface of the temperatures were minimum in Pudanagar Lake and Guntur Lake, maximum in Koraya River. pH was minimum in Pudanagar Pond and maximum in Gundur Lake. Dissolved oxygen were minimum in Koraya River, that is 3.21. Let us see why it's then. Nitrate were minimum in Gundur Lake 0.4 and maximum in Koraya River 3 mg per liter. The ammonia were minimum in Pudanagar Lake 0.15 and maximum in Koraya River. Phosphate were minimum in Pudanagar Lake and maximum in Pudanagar Pond. Table 2 shows a diversity of the different zooplankton. Maximum diversity was recorded in Pudanagar Pond, 11 species, and minimum species, that is 6 species, were recorded from Koraya River. Table 3 shows density and percentage composition of zooplankton in different water bodies. Maximum density of the zooplankton was recorded in Gundur Lake, which is located in the Tiruchirappalli, and minimum density was recorded in the Koraya River. This is also located in the Tiruchirappalli city. A moderate, that is 41 individuals were recorded in Pudanagar Pond and 20 individuals were recorded in Pudanagar Lake. Plate 2 shows some of the common zooplankton species of different water bodies, among which Brachiones caliciflorus and Brachiones caudatus were the predominant species in rotifers. Moina micrura of the Cladocerians was a common species in Cladocerians. In Calanide, Heliodiotomus cinctus were the common species, and Cynodiotomus indicus were the common species. In Cyclopod copepod, Thermocyclops decipiens were the common species of the all the stations. In the present study, there are 17 species belonging to the 5 groups of zooplankton were recorded in 4 stations. Minimum diversity was recorded in Pudanagar Pond, maximum species was recorded in Pudanagar Pond, 11 species, and minimum 6 species was recorded in Koraya River. In general, the rotifer Brachiones caliciflorus, Cladocerian Moina micrura, in Astrocard, Hemicypris species, Calanide, Heliodiotomus cinctus, and Cyclopod copepod, Thermocyclops decipiens were predominant in all the stations. It was interestingly noted that absence of Calanide copepod species in certain stations, whenever rotifers, particular Brachiones species, are abundant, is an indication of sewage pollution. Particularly, Brachiones species are abundant in sewage nearest station, particularly in Tiruchirappalli River, which receives untreated domestic sewage from Tiruchirappalli city, which shows maximum number of Brachiones species. That is indication of sewage pollution. And moreover, there is a village which is nearby the agriculture land. They are discharging simply agricultural wastes into the Koraya River. That is also one of the reason for maximum number of brackish species. Overall density of the zooplankton ranges from 18 individual to 61 individual in 4 stations. However, the minimum density was recorded in riverine habitat, particularly Koraya River, that is 18 individual per liter, whereas maximum density was recorded in Gundur Lake. It is also located in the same city of Trichurapalli, but it is receiving the sewage input. That's why maximum number of individuals are recorded in Gundur Lake. In general, maximum percentage composition of the zooplankton was occupied by cyclopod, 33%, cladoceran 19%, rotifers 8% overall 4 stations. This results clearly indicate that density of the zooplankton is not depend on the habitat. It is depend on the amount of nutrition input. Koraya River and Gundur Lake are located in the Trichurapalli city, which is the city receives more untreated sewage. That might be a reason for maximum density of the zooplankton. Whereas in Pudnagar Lake and Pond, which is free from city, that is, it is located in the Pudnagar village, that might be a reason for least density with a rich diversity. In conclusion, investigation concluded that Pudnagar Pond and Lake has good water quality, which is free from domestic sewage and industrial activities, whereas the Koraya River and Gundur Lake received more untreated domestic sewage from the Trichuri city and industrial effluent from Vanaspati oil, which is located in the Karumandapam region, which is receiving untreated sewage from Vanaspati refined factories. That may be reason for higher density in the Koraya River and least diversity. So therefore, a regular restoration must be taken before discharging these input into the river and increase the zooplankton diversity as well as the other aquatic biota of the particular ecosystem. With this, I conclude my presentations. Any clarification you can ask. And before that, I would like to thank my research supervisor and guide, Dr. K. Altaf sir, for providing an opportunity to present in this wonderful sessions.
Nice work, Dr. Janaki Raman. Any queries? There is one question, sir.
Yes, madam.
How frequent the diversity study has been carried out? Is it seasonal study?
Actually, it is a preliminary study. It's for a MSc project. Due to limited period, I couldn't take my regular sample collection. Actually, this assessment mainly not enough to assess current ecological status. At least seasonal, unless once in a year, regular sample, at least 12 sample in year, it might be good enough to assess the ecological status. However, based on the presence or absence of the zooplankton species, we can conclude what kind of planktons are available, what kind of nature based on the correlation of water quality along with the plankton species. We can conclude. Unless regular sample might be good enough to assess concrete result, madam.
Thank you, sir. Next, I will call upon Ms. Namrata from Banari Amman Institute of Technology. Ma'am, are you there?
Yes, ma'am.
Please share your slides.
It's visible, ma'am.
Yeah, it's presenting. I request the participants to finish the presentations in 5 minutes.
Okay, ma'am. Hello, everyone. First of all, I thank ANET deemed university for organizing this conference. Myself, Narmada K, from the Department of Food Technology for Banariamman Institute of Technology, gonna present about aquaculture, nutrients, and fish feed. Aquaculture is basically the raising of aquatic animals, mainly fish. Fish is considered to be the most important animal and a poor human protein food due its low cost. Nutrition and food play an important and vital role of the continuous development of fishes, and therefore fertilizers and feed research continue to dominate the aquaculture needs. Creating sustainable feeds using quality and low polluting nutrients. Nutrients and diet play an important role in the continuous development of aquaculture. So, this paper will review a number of specific issues in the fields of aquatic animal nutrients and feeding that are critical for sustainable aquaculture production. Important feed Important feed composition and better feed efficiency will result in higher fish production, low cost, and low waste production. This is the reason for fish nutrition and feeding play important role in the development of aquaculture. Fish is filled with omega-3 fatty acids and vitamins such as D and B2. Fish is rich in calcium and phosphorus and great source of minerals such as iron, zinc, iodine, magnesium, and potassium. Our body don't produce omega-3 fatty acids, so we must get them through food we eat. Some potential alternative ingredients are that this can be used as fish feed. Not only this, farmed seafood has significant growth potential as a source of food and fiber for both aquaculture feed and human consumption. So natural food, supplementary feeds, complete feeds. There are three types of feed used in fish ponds. Natural food. It is naturally in the pond. It may include detritus, bacteria, plankton, worms, insects, snails, aquatic plants, and fish. Their abundance greatly depends on water quality. Liming and fertilization, in particular organic fertilization, can help you to provide a good supply of natural food to your fish. The next one is supplementary feeds. It's regularly distributed to the fish in the pond. They usually consist of cheap materials locally available such as terrestrial plants, kitchen waste, or agricultural feeds may be also regularly distributed. They are made conform a maximum of carefully selected ingredients to provide all the nutrients necessary for the fish to grow. They must be made in a form which the fish find easy to eat and digest. These feeds are quite difficult to make and form and are usually quite expensive to buy. The system of production can be defined according to the type of food given to the fish. Extensive, semi-extensive, intensive. Extensive. Fish production depends entirely on natural food. Semi-intensive. Fish production depends on both natural food and supplementary feed. More fish may be reared in the pound. Intensive. Fish production depends entirely on complete feed, and the stocking rate no longer depends on food availability but no other factors such as water quality. Nutrition and feeding influence the growth, reproduction, and health performance of fishes. Feeding the fishes with nutritionally enriched feeds may dramatically increase the overall production. So, nutrition is one of the essential area to be focused by aquaculture industry. Many researchers are currently undergoing towards the development of cost-efficient feed for engaging the fish and shrimp production. Replacement of fish meal with various plant-based ingredients will be superior way to reduce the cost of fish feed without changing the nutritional profile. There are several plant-based ingredients like soybean meal, sesame meal, cornstarch, etc. could be used to replace fish meal in the fish diet. Though fish convert feed to human food very efficiently, the feeding cost of production need to be controlled. Feeding fish require an understanding of the progress of digestion, the digestive system of fish nutrition. Fish consume feed for energy. They use the energy for growth, activity, and reproduction. In this fish diet, feeds containing protein, fats, and carbohydrates supply energy. Protein in the diet also supplies 10 essential amino acids, and fat in the diet supplies essential fatty acids. Fat soluble and water soluble vitamins are also supplied by the diet. Minerals are supplied by the diet and by the water. With a great understanding to the nutritional aspects, it is very much possible to make feed nutritionally balanced. so that higher growth, best food conversion ratio and less polluting to environment could be achieved. So now we have came to the end of the presentation. Thank you giving for this wonderful opportunity to us. Thank you.
Nice presentation, Ms. Narmada. Any queries? There is no queries. Okay. Next, I'll call upon Mr. Deepak from Banari Amman Institute of Technology. Mr. Deepak, you're there?
Yes, ma'am.
You can share your screen.
My screen is visible, ma'am?
Yeah, it's presenting. You can skip the introduction part.
Okay, ma'am.
Okay.
First, let's see the aim and objectives of our topic. As a global aquaculture production increased to meet the increased need for the food and especially seafood, there is an increasing demand for feed supply. The main objective is that feed management and non-farm practices relating to feed, therefore, an essential part of managing not only economics of farm but also flow of nutrition from raw materials into the fish. The final product should be available in edible and attractive form to humans. Then comes methodology of our study. The efficiency with which fish converts their feed into body weight, the study fall into following broads. Categories: feed, manufacture, logistic and storage, on-farm handling, and feeding methods, timing and quality. Here is a short discussion on proposed topic. Fish recommends versus dietary specification. The nutritional requirement of a fish vary mainly depending on the species, its size, or its life and environment. For example, Atlantic salmon requires dietary pigment to make the flesh red. Next comes the feed raw materials type and quality. Different raw materials have different nutritional value and also they are prone to contamination, so it's important to check for those quantities. Here comes the important thing, technical properties of feed. They include impact of cooking, density, hardnesses, moisture, content, pellet size, and so on. The slides displays various forms of fish and its quality. Here is the brief description of fish feed quality and trash or a low value fish, then the mashes, followed by mines and pellets, the important feed of fish. This displays the cooking process, those feeds which important in determining the carbohydrates. Then comes the brief note on important note for the feeds, its density, followed by the hardnesses of the fish feed and moisture content present in the feed. Here is the note on pellet size and the small dust particles present in the fish feed. Here is the important factor in manufacturing, that is on-farm action storage. This must ensure proper storage facilities and pest control measures. Here is the picture representing storage site of fish feed products. Then comes the tool to measure feed and feeding performance with economic conversion ratio growth rate with a simple formula. There is the cost of production, which includes input cost, labor cost, and so on. Here is the very important factor that understanding nutritional requirements is splitting protein and lipids, followed by the carbohydrates. Then the vitamins are classified into water soluble and fat soluble in feeds. The another important nutrient, mineral, usually obtained by fish in water. Here is the type of feed, that is wet moist feed, depending on their texture, followed by the dry feed varieties. Then comes the product quality feed, that is medicated and hormonal feeds. This slide shows the method of feeding the fish. Then comes the time feeding, that when to feed and when not to feed. It is determined by the nature of fish and oxygen demand. Then comes the rate feeding, that is how much do we need to feed. Here is the important factor that other feeding is the management of feed waste, which usually occurs. Here is the representation of understanding nutritional recommends starting from ingredients to packing of feeds. Here is the note on grinding and its feed grinding machine, followed by the process of extruding. Then comes the process of mixing and ingredients of the feed. The final process of drying, weighing and packaging of products. This is the flow chart represent aspects of creation, use and revision of feeding and feeding management plan for growing fish. Here is the result and conclusion of our topic. Feed is the major product in aquaculture, both in terms of quality and quantity. In order to get the best result, certain measures have to be undertaken, such as feed fit for a purpose, feed delivery securely to the farm, feed store on the farm in manner that will maintain quality, healthy fish, and optimum environment. Accurate knowledge of the number of a fish in each unit or the size. Knowledgeable feed and requirements. I thank you everyone patiently listening to my presentation. I thank this conference organizing team for providing me and my team member for this opportunity. Thank you.
A good presentation, Mr. Deepak. Any queries? Oh. Okay. So next I will call upon Ms. Ashtaya from Central Institute of Fisheries Education, Mumbai.
Yes,
Ms. Ashtaya,
yes,
please
I'm
share
sharing.
your screen.
Yeah, I'm sharing my screen. I hope my screen is visible.
Yeah.
A warm good afternoon, everyone. Today, we are going to time travel for a while to examine the past and present, as well as to explore the exquisite future of aquaculture nutrition. Aquaculture and nutrition are inseparable entities. Each have to work hand in hand with each other. Without further delay, let me cut to the chase and straight away jump to the topic of discussion. And as far as nutrition is concerned, it's a complex and meticulous branch of science. Let me give you an instance here. For example, there are 139 crore population in India, and even the all population do a research in nutrition, I feel it's not enough. Because if you change the inclusion level of protein from 20 to 35, or simply you change a single ingredient that is used in feed formulation, all these subtle changes can create a remarkable difference here. The image what you're seeing here, first you will think that it's frivolous and nonsensical one, but it's our own creative model. And we have named this model as bicycle model to make even a lame person understand how these things are interconnected. So the first wheel, it represents the nutrition part, like the feeding strategies and the feed that are used in culture. And the back wheel, it represents right from the stocking densities, type of species used, water quality management, everything will be covered here. So you may even think why we have chosen a bicycle. We might have gone for a fancy Pulsar or a Lamborghini, anything of it. But it's like we need to emphasize something strong here, that we need to maintain the system sustainably. And you also know that aquaculture is not a single take. It's a slow and a steady process. And the person here is the farmer, and he need to balance these two things continuously, right from the nursery to the harvest. When he manages these two things properly, then there is no invitation through a special person named Mr. Disease. So this is just the global chart representing the animal feed production sector, and you can see that broiler stands first. When you take the global aqua feed production, it is almost around 49 million metric tons, whereas the India aqua feed production is almost around 2.5 million tons per year. But whereas the fish meal inclusion is exponentially high in aqua feed sector. So because of continuous usage of fish meal, we are actually dredging our oceans empty. And the second most ingredient used is soybean. People think that soybean could replace fish meal. Actually, you must know that soybean is even deficient in certain amino acid, and they are the major reason for the deforestation of rainforest. So you may think that if these things are happening, then we are definitely on a dangerous pinch point. I feel enough is enough. It's time for reshaping. That's where our rescue person comes. Yes, I'm talking about black soldier fly. So I would like to call this as a brown live gold. You also know that insects are natural and they are normal one. Insects are actually nature's perfect and they're even upscaling machines. Actually, there's a huge disconnect between what we eat and what we feed for the animals. All animals, including the broilers or fish, they need nutrients, protein, and everything like we do. So what we are going to do? Hence, the black soldier fly, it's mainly renowned for the ability to consume the organic waste and they are harmless naturalized species. So what happened here is each female, they can almost lay around 1,000 eggs and we need to incubate these eggs in an organic substrate. After that, larvae will be hatched out and these larvae can very well munch on organic waste. This organic waste could be anything, right from the kitchen waste, vegetable waste, or cafeteria. It could be anything of your interest. And it's not just about the black soldier fly production. Here we are concerning about the waste management as well. Almost third of all waste produced in the world is actually from the food. So this larvae can very well make use of the organic waste and after once they hatch out that larvae, after a particular period of time, we can harvest them and transfer them as an insect meal. Later, we can use them for a feed formulation. Later, they will be developing into pupa, adult, and the life cycle goes on. So there is 365 days you can produce this and at any climate you can produce. This is the major highlighting point. So at the end, we will be having two most important things. The first thing is regarding the organic protein. If I'm talking about the protein, you must also know that it's not about the crude protein content. It's mainly about the essential amino acid profile. And this BSF is mainly efficient in certain amino acids like lysine and methionine, and which is deficient in certain novel plant-based ingredients. As well as they have a good amount of crude fat from 10 to 30%, as well as they are also rich in calcium. So whatever left over is the compost. So this compost is even rich in nitrogenous matter and there is a compound called frass. It is actually this bug excreta. So what happens is you can use this as an organic fertilizer for agriculture and horticulture. So you need to notice something here. The end point of one industry is used as the starting point of other industry, which means circular economy is achieved. We don't stop here. We still have something more. We can even get some lucrative biomaterials in the form of chitosan, melanin, probiotics, etc. And also there is no zoonotic infection as of now. And it has overall, the research is happening all over the world and they have found a tremendous result in case of shrimp, bluegill, and also in tilapia. So I would like to summarize by saying that our ICR SIBA has already taken an initiative by tying up with a startup that could produce a BSF. So I have a strong conviction that success is not very far. It's highly achievable. Only we need to focus on diversification, intense research, as well as the mass production. We can definitely bring laurels to this industry. So my take-home message is, remember guys, yesterday's waste is today's wealth. And you must remember that you miss 100% of the shots you never take. I'm leaving this to you completely. It's our planet, so you can either choose to live sustainably or sabotage it. Think over it, guys. Thank you, everyone, for this understanding.
Nice presentation, Ms. Astaya. Any queries? Okay. So next I'll call upon Ms. Niveta from Banari Amman Institute of Technology. Ms. Niveta, please share your screen.
Is my screen visible, ma'am?
Yeah. Please continue.
Good afternoon to all present here. Thank you for giving this wonderful opportunity. Today I'm going to present about biotechnology and its application of aquaculture in fisheries. I'm Niveta A. I'm pursuing second year biotechnology in Banari Amman Institute of Technology. Biotechnology provides a powerful tool for sustainable development of aquaculture fisheries as well as food industries. Increase in public demand of seafood and decreasing natural marine habits have encouraged scientists to study the way that the biotechnology can increase the production of marine food products and making aquaculture as a growing field of animal research. There is a growing demand for aquaculture. Biotechnology can help to meet the demand. Successful development of application and biotechnology are possible only when there is a broadcast research, variation, breeding agronomy, psychology, pathology, biochemistry, and genetics of manipulated organism exist. The potential area where biotechnology in aquaculture include the use of synthetic hormones, induced breeding, transgenic fish, gene banking, uniparental and polyploidy population health management. The main aim of this paper is to see how biotechnology is applied in aquaculture. Biotechnology in fish breeding. Gonadotropins releasing hormone is now the best available biotechnology tool for inducing breeding of fish. GnRH is a key regulator and a central inhibitor of production cascade in all vertebrates. It's an decapeptide and was first isolated from pig and sheep hypothalami, which is ability to induce the pituitary release of a leucating hormone and fossil stimulating hormone. The most recent GnRH purified and castrated was Crosfield and Robinson. Depending on the structural variant of biotechnology activities, number of chemical analogs have been prepared, and one of them is a salmon GnRH analog profusely used now in fish breeding and marked commercially under the name of Ovarim throughout the world. The induced breeding of fish is now successfully achieved by development of GnRH technology. Next, we are going to see about transgenesis. Transgenesis or transgenic may be defined as introduction of echogenous gene or DNA in the host genome, resulting in the stable maintenance, transmission, and expression. And the idea of producing transgenic animal became more popular when Palmiter et al. First introduced transgenic mouse by introducing the metatholanines or human growth hormone fusion gene into mouse egg, resulting in dramatic increase in growth. The technique has now successfully applied in number of fish species, and increased resistance of fish to cold temperature has been another subject research in fish transgenics of past several years. Cold water temperature poses considerable stressor to many fish, and few are able to survive water temperatures such below 0 to 1 degree Celsius. The tool of the future transgenic fish production is development of embryonic stem cells technology. Although significant progress has been made several laboratories around the world, there are numerous problems to resolve for the successful commercialization of transgenic brook stroke aquaculture. To relieve the full potential of transgenic fish technology in aquaculture, they include more efficient technology of mass gene transfer, targeted gene transfer technology such as embryonic stem cell gene transfer, suitable promoters to direct the expression of transgenes and optimal levels during the desirable development stages, identification of genes and desirable traits of aquaculture and all applications, information on physiological, nutritional, immunological, and environmental factors that maximize the performance of transgenes. Safety and environment are main. Biotechnology and food health management. Disease problem major constant for development of aquaculture. Biotechnology tools such as molecular diagnosis method, uses of vaccine and immunostimulants are gained popularity for growing the disease resistance. Biotechnology tools such as gene probes and polymerase chain reaction PCR are showing the great potential in this area. Gene probes and PCR based on diagnosis method have been developed for number of pathogens affecting fish. In the case of finfish aquaculture, number of vaccines against bacteria, virus have been developed, and some of these have been conventional vaccines consisting of killed microorganisms. In the vertebrate system, against the disease is common strategy. Among the recent studies, have shown that non-specific defense system can be stimulated using microbial products such as lipopolysaccharides, glycans, or glycans.
Ms. Nivedita,
Yes,
can
ma'am.
continue.
Among the immunostains known to be effective in fish glycans or limazole enhanced pathogenic activities or specific antibody responses. And the next topic is cryopreservation of gametes or gene banking. Cryopreservation is a technique in which involve long-term preservation and storage of biological materials at very low temperature, usually at minus 196 degrees Celsius, the temperature of liquid nitrogen. The technology of cryopreservation of fish spermatozoa has been adopted for animal husbandry. The first success in preserving fish sperm at low temperature was reported by Blaster, who fertilized Heron's egg with frozen Taiwan semen. The spermatozoa of almost the cultivable fish species has now been cryopreserved. The most of the plant varieties that have been produced are based on the gene bank collections. Aquatic gene bank, however, suffers the fact that at the present is impossible for cryopreserve only male gametes of finfishes, and there is no viable technique of finfish egg or embryos. Therefore, it is essential that gene bank of cultivation and cultivable aquatic species. As a conclusion of my presentation, biological research and development are growing at a very fast rate. The biological has been assumed greatest importance in recent years in development of fisheries, agriculture, and human health. The science of biotechnology has endowed us with new tools and a tremendous power to create novel genes and genotypes of plants, animals, and fish. The application of biotechnology in fisheries sector is relatively practices. Um, the increased application of biotechnological tools certainly um, certainly revolutionize our fish farming beside in biodiversity conversion. The paper briefly reports that current progresses and trust areas in transgenius uses of synthetic hormones in fish breeding, biotechnology in health management, and gene banking. Thank you.
Nice presentation, Ms. Nivedita. Any queries? Okay. We will continue the next session after the lunch break, and the participants are requested to join the next session at 2 p.m. using the same link. Thank you.
Thank you. Thank you. Let us join sharply at 2 o'clock. Thank you. Sindhu sir, any announcement?
No, sir. Robert, very clear. The participants can use the same link to join the meeting.
Oh, yes.
They can start the meeting by 2 o'clock.
Oh, yes. Yes. Thank you so much.